we'll have to send people the link in order to
Okay,
yeah.
good.
Okay, I'll get the chat open. I'll get the Q&A open. I can keep an eye on those. So, oh, hello.
Hello.
Right, we're now live streaming. Let me grab the link. I will post it in the chat to all panelists and attendees.
Yeah,
I will send that to Dan. So we don't have Dan yet.
Oh.
I don't think. Or do we?
Watch. What is this one? KJ.
Just to check, can people hear me?
Yeah.
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 Okay.
Yeah.
Awesome.
Okay, let's see. Yeah, it's live streaming. Super, looks good.
Okay.
Perfect.
I'm going to start our cheesy music.
Aw.
Ah, it stopped, hasn't it? I'm just going to skip ahead a minute. So I realized I cannot click away from the presentation, otherwise it will pause.
Occupied for a lecture.
Yeah. Do any of the panelists know if there's a keyboard shortcut for mute, unmute in Zoom? I see puzzled faces. Have to look that up. Alt A. For those of you who are just joining, thank you for waiting. We'll be underway very shortly. And we're very glad to have you here. Welcome to the Atom Probe Technical Committee meeting on software for enabling research. It's great to have you all here. We're starting off with a short introduction to the Atom Probe Technical Committee, and hopefully that gives us a little bit of a buffer for those people who aren't quite on time, still figuring out time zones. Well done for those of you who have joined on time. It's nice to have you. For our panelists, just a reminder to keep yourself muted if you're not presenting to avoid interference and to try to keep to time. For everyone else, please use the Q&A function that you can find in your Zoom clients to ask questions. We will repeat these questions to the speakers and they can answer them live, or after the talk they can write text replies as well. We'll try and answer at least 2 of these questions per talk. We have a dedicated time for discussion at the end. So on the right, you can see our little program. We've got half an hour. That says an hour. We've got half an hour at the end. You're welcome to stay for discussion. The meeting is being recorded and we plan to release the meeting video publicly. So please bear that in mind. And after the meeting, within a few days' time, we'll be sending around a link to that recording or recordings if we split it up, and a list of the different software discussed, and any links to interactive websites or repositories or whatever that will come after that. Right now, I'm going to hand over to Dan Haley, who's going to introduce the technical committee as soon as I can give him remote control access. There we go, Dan.
Thanks very much, Andy, and it's glad to see everybody here. We've got quite a lot of participants joined us from all over the world. So it's really great that we can host this workshop and that we've all decided to attend. So I guess the first question that I sometimes get asked is, what is the Atom Probe Technical Committee and who are you guys? So to me, that's an interesting question. So we really initially started a bit of a while ago, and we started the last APT&M meeting, and there was some questions about software and how we can try and support research within the community. So the people who form that committee were myself, and we've got 8 members from around the world. We've got people from Sydney, from Germany, we've got people in the US, France, we've got people from industry, and we're in it for 4 years each, and we're trying to basically work out how we can support the activities that are underway in research throughout the world at different labs to try and find a sort of common way to join what we're doing. So we were initially appointed. We had basically a support letter where over 30 labs from around the world, or around 30 labs from around the world, supported our action of trying to form a committee to support this effort. So we were initially, as I say, appointed, but we're elected within the APT&M meetings, and our positions are 2 + 2 year positions, and we meet sort of every 3 months to discuss sort of activities within the community and how we're going to go around supporting this. So, I mean, our goal of our committee is to try and provide guidance to the community. We're not a research group. We're just trying to see what commonalities are coming out and how we can try and help researchers with their work. So we really operate under theme according to our charter, and our current theme is the enabling and enhancing a computational analysis of atom probe data. So our aim is to try and, in this theme, is to try and improve computational analysis, interoperability between labs, and primarily reproducibility. We report to the IFES steering committee, who are the people who set that theme. And so what we're trying to do is avoid the situation where, you know, someone says, okay, we've got to do something, we've got to create new standards, we've got to create new ways of doing things. And oh, look, now there's 15 competing standards. No, we're a cross-community effort that is drawing from around the world. that is really trying to centralize what we're doing and support that. So how and what do we do this? So we have regular virtual meetings, and there's a public email list that you can see the link there for. So all the activities that we undertake, or almost all the activities that we undertake, are publicly available and are conducted publicly. So anyone can join this meeting and you can review what we've done. You know, we often will say we'll take comments, suggestions, requests, either in person from your local TC representative if you have one, or we can discuss them by email, and we can bring these questions from the community up at the meetings. So as I say, we're trying to provide common research frameworks, documentation, and infrastructure for researchers. And we really achieve this through a couple of key tools: standards, reference implementations, and documentation. So we're trying to provide sort of baseline levels that people should be trying to achieve in terms of standardization, in terms of how you present research, things like this. So these are the kinds of things we're covering. So the last thing that I should mention is FAIR. And this is really at the heart of what the TC is trying to do. It's trying to be improving findability, accessibility, interoperability, and reusability, which forms the sort of four cornerstones of the FAIR initiative. So what is FAIR? It's a shift to try and move from closed data and research towards more open and network scholarship. And this is something that, if you're a junior researcher, this is going to be very important throughout your career as more and more countries start to mandate this. So if you're in the EU, if you're in the UK, if you're in the US, under certain funding schemes, you'll find that this has actually started to be mandated. So what it means is that some of your scholarly publications have to now be open access. Your data also needs to be findable and reusable. So the FAIR initiative is to try and improve open research outcomes, to try and share primary data, try and share things, maybe even samples that you might be having, and provide this to the community. And so there's really a big question about how does one actually do this? I think the only way we can achieve this is to come together as a community and decide what is the method by which we're going to do that. And the technical committee is at the corner of trying to answer the technical questions of how do we go about doing that. So I guess at this point, I'm going to hand back to Andy, who's just introduced our next speaker. He's going to start discussing some of the initiatives that are available throughout the community. And at the end, we're going to have a bit more discussion about what the technical committee has actually been doing for the last couple of years and where we're going in the future. All right, thank you.
Thank you, Dan. That's great. Excellent introduction to the TC and why we're all here. We've organised the meeting so that we have these three invited speakers speaking first. And some of these are a mixture of video and slides. I hope that that's amenable. This is what works best for our presenters. We do have almost all of the speakers here live for answering questions. So feel free to ask as many questions in the chat and the Q&A as you like. And like Dan said, there'll be time at the end. We have a short presentation on some of the TC's activities and then a space dedicated for discussion. So save your TC questions till the end and we'll have an open forum for discussing them there. So next we have a presentation on the data streams and visualization that Patrick Stender has been working on.
Hello and welcome to my talk about the APT data streams and visualization as we handle them here in Stuttgart. My name is Patrick Stender, and it's my pleasure to show you briefly what we are doing at the moment. As you may know, here in Stuttgart, we are developing and operating our own custom-made APTs. As some other groups across Germany and also in Japan are still doing. These custom-made APTs are controlled with our software called Tab Control. Mostly these instruments have been developed over a decade of years, so the technology has changed and there are different interfaces available controlling the software, so our software had to adapt to different hardware. The software itself is based on C++, has a Qt cross-platform compatible user interface, and the main goal is to control the hardware during a measurement process and to store all data necessary for each atom detected to create a data set, which we call WAR file for later reconstruction. As mentioned before, a lot of data has to be recorded during the measurement process, as you already know. So we have to determine the voltage applied to the tip in the moment the atom leaves the tip. We have to control the laser position onto the tip. We have to measure the time of flight signal and some other data as well. From all the data coming from preamplifiers and other sources of data, the measurement software is evaluating the incoming signals and determine valid events and storing them with the respective information in the so-called, for us called, raw data format. So here you see a snippet of the definitions. So we store the necessary information like the voltages applied. If you need the reflectron, you also store the voltage set to the reflectron, the detector impact position, the time of flight, a time indicator during the measurement, and the pulse window, which is identifying multi-hit events for later multiplicity evaluations. For the later reconstruction, a machine parameter data is necessary. As mentioned before, all atom probes slightly different construction, so therefore we need a so-called machine parameter file. The machine parameter file includes the detector area, detector efficiency, and other variables which have been determined by calibration of the instrument. So this parameter file is unique for each instrument. The file itself is written in JSON, so human readable, and the parameters depend on the respective construction of the machine. Equipped with a raw file and the parameter file, you would leave the laboratory and use the software which we call Skeeto to reconstruct your data. This is currently in the version 2.03 and still under development, so features are added and bugs are removed. All algorithms are C++ based. For the visualization, we use OpenGL. And for the UI, we use Qt. So the whole system is cross-platform compatible. Currently, we are just using a Windows version, but Linux and Mac version are also possible. Initially, you would create a project file containing not just the two files I mentioned before, the raw and parameter file, but you would also include an alloy file, which gives you the definition on which atom should have this nice yellow color or blue color. I will go into detail to the alloy file later. If you have different sources of data, you can also load data from these, which includes tabsim files directly, POS files, EPOS files, and also ATO files, and some other. The software itself is built in a way that it guides the user through the reconstruction process. So it's a stepwise procedure. So you start with the project, where you create your project file and determine the source of your raw data. So here you have all data available. The next step would be the raw data file inspection and filtering if necessary. So you can filter according to the data range, the voltage range, the radial range, attitudinal range, or whatever you would think about by creating a JavaScript which you can add into the pipe by determining which parameters you want to get into your script. A skeleton is created, you can add your code, and data coming out of your code is fed back into the data stream. So therefore you can also create custom scripts, which is quite convenient for students to get quick solutions for a few problems. After you have inspected your data and are happy with it, you would calculate the respective mass spectrum, and depending on whether you already have an LFI defined, you can move the mass windows, which you need for color coding, just by drag and drop to the correct position, and check your data and identify your data as you need. For the alloy file, we are using also a human readable JSON file, which contains definitions of elemental species, so just pure atoms. There are some values stored for each element, and molecules are made out of these elements, giving a new definition in the molecule section. And last but not least, you would determine in this file the respective window from which position to which, for which element this window should be available. Of course, you can edit this file by hand. For a lot of mesh windows or species inside your measurement, this could be quite tricky or tiring. Therefore, of course, the software contains the necessary information to help you through that. So all elemental data are stored in a database, on an SQL database server. So you just click them and they are added into your alloy file. And you can also create molecules by clicking and then determine the parameters necessary, like density, constituents, and so on. If you are happy with the definition, you can also store them locally for your use. And if you are a very nice, kind person, you can also upload this to the SQL server so that anybody that is searching for a special name or a short name can have a look what's already stored for this respective element in the database. So they can reuse your definition of molecules and, of course, download them and, if necessary, change the parameters again for local use and store a second version if you are not happy with the first version onto the server. After having these definitions, of course, the reconstruction is quite important. For the reconstruction, we are using different algorithms based on published papers. The respective parameters are given here. Of course, the definitions, pseudocode, and code snippets are available online if you want to check whether the implementation is correct, or if you want to add something new, you can also add a custom script. So you can think about your own reconstruction algorithm and test it out, either just changing small aspects of the given ones, or just imagine your complete new reconstruction scheme and testing it out. Of course, we are adding different kind of new algorithms if published, so that this is under constant change. You have all necessary information to verify if your reconstruction is correct according to the respective reconstruction algorithm. Finally, you want to visualize your processed data. You can imagine that the data pipe in this direction is ending here in the so-called scene point. All the data available here can be further processed by adding different kind of modules. In this kind, for example, an element filter. This element filter, when clicked, gives you the options which are unique for the respective module. And then you can change or alter the data going further the stream. Of course, you can also branch the streams to get different branches for evaluation if you want to analyze different aspects of the tip at the same time. And by this, you can be very flexible in manipulating the data the way you want or evaluating the data you want. Of course, there are different tools available. These different tools include various filters and analysis tools like cluster search, isosurface, autoslice, composition profile, and so on. Having all these data created, of course, it's very important to export these data to known file formats for further processing with third-party scripts, software, and so on. Therefore, we offer quite a lot of predefined export functions, but you can also, from the whole data set, export your own file depending on the values you want to export in just a text or ASCII file. This was very quickly and briefly the way how we deal with our data. If you have questions, of course, I would be very happy to answer them. Please contact me by any possible way. And if you have questions now, I would be very happy to answer them. Thank you very much for your attention.
That's great. Thank you very much. I believe we have Patrick on the line, as it were,
Yes,
to answer questions. Hello. Good to hear from you. Sorry, my... where are we? Hello. At the moment, I don't see any questions in the chat, but I had a quick question. How sort of interoperable is your software? Can it be used with can we open up any raw data file and run it through the same software, or is it only for the sort of raw format that you have from your atom probes?
Of course, we developed it for our file format initially, but getting into contact with different groups, there was always the question how we can share data. So from all code we could get, we try to implement also functionality to import them as EPOS files, POS files, and so on. Not all files give you the full possibility of analysis because some data are missing. So if we would have something that would be generalized, of course, this would be a big benefit to be implemented.
Okay, great. We'll just hold on a couple of minutes in case we have any more questions. How accurate is the time since epoch defined to be? Could you correlate hits from different laser pulses with accuracy? I think this is related to whether you only have second accuracy or whether you need milli, micro, nanosecond accuracy, etc.
Yeah, using 64-bit, we have milliseconds accuracy with the epoch time, but we have this pulse window indicator, which gives you a unique identifier after every laser pulse. So you can decide in between every laser pulse. The time is just more for getting quick data on how the measurement developed over a long time of period.
Okay, thanks very much. Does the software have the possibility to take into account peak overlaps in the mass spectrum?
We are working on that actually, not yet, but we are going to implement this in the near future, yes.
And there was a sort of internal question. How do you see the technical committees work as someone who's developing atom probes yourself?
Oh, I think it's very positive. As I said, it would be very nice to get more, at least definitions of raw data or a unique raw data file set, which could be exchanged between different groups more easily. So there's always a bottleneck how to get the data from one group that has a different machine. I know we are just a very limited number with exotic machines, but it's still, for us, it would be very helpful.
Well, it's funny you mentioned that because I think we'll hear a little bit more about that later on. One last question. Is the software available outside of your group for other people to test and try, or is it closed at the moment?
Yes, yeah, of course. We would be happy to give out test versions. We are still under development, so any feedback would be great for us. So if you would like, you just give us a short mail, and me or my colleague would send out a version account so that you can test the software.
That's great. Thank you very much, Patrick.
Thank you.
Thank you. Tim, if we have you on the line, as it were, I should have given you remote control of the screen and you should be able to go ahead.
All right, can you hear me?
Yes, I can hear you just fine.
All right, fantastic. So thank you, IFE's technical committee for having me speak today, and thanks to David Reinhardt for volunteering me and letting me out of my cage to talk to you guys today. So I'm a lead engineer at CAMECA. I've been there 18 years now, starting with Imago and now CAMECA. And currently I'm the architect for our new AP Suite software, Atom Probe Suite software. We just released our 6.0601 version. And I'm going to talk about one of the new features in 601, which is custom analyses. So moving right along, let's see if I can advance the slides here. All right, so it's both my philosophy and CAMECA's philosophy that we want to embrace the Atom Probe community's independent efforts for running Atom Probe analysis software. As you probably all know, there's been a long history of people running independent programs. I've been to lots of talks where people are doing the same thing in different languages in different parts of the world, and there's not a lot of sharing going on. So we're, along with you guys, of course, the technical committee, hoping to try and pull in some of that effort online so we can all leverage each other's work. So the first thing that we've done here is made a new feature in 601 called the custom analysis. So this is what we have right now. it is just a first start into this world of embracing external analyses. If we waited till it was perfect, of course, we would never release anything. So we really are excited to get this out in the world and get some feedback. So this new custom analysis feature is a custom module. It's a DLL that you build, and the software loads it on startup and adds new menu items into your analysis tree. So you right click anywhere in the tree like you normally would for adding nodes. You can find new custom analysis nodes in the list. Those are built at startup from analyzing the modules that are available. And the modules then, these custom analysis modules, have access to the data in your reconstruction. So that would be the raw ion position, XYZ, mass to charge ratios, the ion type, the identified type, which would be, say silicon or iron, and also the range data which is used to generate those ion types. And because these modules are just normal DLLs, you can call any external code you like. You could call a Python in an external Python script, all quite easily. We did try, actually, in our previous version 6.0, having a raw C# console in AP Suite. And we've actually taken that out in favor of having these external DLLs, because it turned out that editing code, C# code directly in AP Suite was difficult without having all the tools available to you from an IDE, like code completion and inspection. So everyone was using Visual Studio anyway to edit their modules. So we decided just to make it fully into the Visual Studio world. So you can still add a C# console. There's a special type of external module. You call it, it's called a, if I remember correctly, a custom analysis with console. And then you can have your C# window if that was a way that you'd like to do it before. So moving on here, here's an example of a custom analysis user interface. On the right, or sorry, on the left side is an automatically generated UI for editing the parameters of your module. All you have to do in your module is list the parameters in a class. It's called the options class, and they will be automatically shown here with the defaults. And then you can, of course, change those defaults if you like. And those values, those parameters, will get saved with your analysis tree or your analysis state. And then when you click run, it calls a function in your module called run that does whatever processing you like. And there's a thing called a view builder that the interface is passed into your run function, which allows you to use the built-in visualization tools in AP Suite for outputting your data. So there is text and tabular data. The example here on the right side is tabular data. In this case, some compositional information. And then we can also have a few different types of visualization. You could have 2D plots, both line and histogram types of plots. And of course, histograms, you can do linear and log, you can change binnings and that sort of thing. And you can have also like a graphical heat map, color map type plot, which is shown on the right there. And you can have a more standard Atom Probe software 3D view with whatever data you'd like. And you can also add points. You can add points and surfaces to that 3D data. And the interface for the view builder is very, very easy to use. It was meant to be very easy for the users to add new visualizations. So those modules that I showed a few slides ago in the right-click menu, all those are in one DLL. The source code we distribute with Atom Probe Suite. It's a zip file that lives on your machine after you've installed Atom Probe Suite. You can see that here. User IVAS custom analysis zip. Unzip that to in the directory of your choice, and you can open that with Visual Studio. There's a solution file in there. You can open that right up and check out all the source code for those examples. The examples are simple, and they provide some good good examples of how to use things. So unfortunately, we do not have at the moment a documentation package for our API for these modules. Again, if we waited till that was ready, we'd never would be late. So we've provided these examples instead. And the beauty of .NET C# code is that you can inspect all the available functions that you can get from our API. So you can use what's called the object browser, and you can see all the various functions you can call. And the examples show example use of those functions. We will have more documentation later, but it's right now the examples are what we have. So after you build, you get a DLL, and all you have to do is copy that back into your Atom Probe Suite installation folder, restart your app, and the module will be loaded, and it will appear in your custom analysis menu. I also want to talk about our APT file format, which instead of sticking with POS and EPOS for our reconstructed data, we've moved to this new format called APT. I wanted to have a format that was expandable. So instead of having a fixed number of records in the data file, now you can add as many what we call sections as you like to the end of an APT file. And the format is very clear, and there's nothing weird. You can use any type of language to read and write it. And the sections are arbitrary. They don't have to follow any known standards. We have our own standards for position, mass, that sort of thing. But you can also add things from your custom analysis, like some new phase information, say, or local density, or something of your choice. Full specification is in our user guide, and I will have this available if you contact me, or you can find it online. Eventually, we're working on having a new home for all this stuff. So this is the exciting stuff. What's coming next? So I said before, this is just a first pass in AP Suite 601 on our next release, which is coming hopefully right away next year. These custom analysis modules are going to get access to a lot more data, so including other things in the analysis tree, like any other analysis you might have, like concentration data. You can also see the 3D concentration grid or isosurfaces from other nodes in the tree. And also you can get access to any type of raw data available. Right now, you can just see kind of your standard POS data, XYZ mass. In the future, you'll be able to see any of the things on the right side of the screen here. Also, we're adding a lot of new visualization options. So you can see arbitrary data from your APT file. So if you add new APT data items, you'll be able to visualize those without having to write your own visualization code. You can also generate data from a custom module, which I think has a lot of pretty cool opportunities. And for things like creating a little mini simulator right in the middle of your analysis tree, or doing some custom filtering. So once you have a custom analysis node, you'll be able to right-click on that and add any normal analysis, like concentration or isosurfacing, like you would any other ROI in our software. And we're also going to have a new type of module, a custom reconstruction module, or custom reconstruction scripts, so you can do your own reconstruction algorithms, or automate some of our existing reconstruction wizard steps and parameters like K-factor. And we're also going to have, hopefully, some sort of online integration where if you have a GitHub repository, you can point AP Suite directly at GitHub and automatically download custom analysis code right to your computer without having to do it manually. And we're hoping we can start to have a online repository for people to submit open source modules where people can share different ways of analyzing their Atom Probe data. So that is all. Thank you so much to the committee for having me. Especially also thank you to GPM for providing some of our examples and helping us define and evaluate our custom module interface. So are there any questions?
Okay, thanks, Tim, very much for an excellent presentation.
welcome.
So should remind everybody to please put your questions. So we've got a question from Ben Kaplan's. He says he can see that TOF and TOFB is on the APT file branches. Can you just speak to what the difference between those two are? I guess he's trying to ask what sort of rawest form of data that you have available is.
Sure. TOF, let me pull it up on my TOF is raw TOF. TOFB, I believe, is bull corrected time of flight. So like flight path corrected time of flight.
Okay, so would you say that the TOF therefore is the rawest form of data that's available from the
Correct. So like the rawest form, if you wanted to do very basic reconstruction, you would want time of flight, voltage, and detector X and Y, quite similarly to the last talk, Patrick's talk.
Okay, thanks very much. I just have a quick question myself, actually. In terms of interchange between different labs, I mean, how does CAMECA see? I think currently people are mostly passing around our HIT files between the labs that have CAMECA-based instruments. How do you see interchange of data going forwards for lab users?
That's an excellent question. Our new software has an export function, which will export all the data from experiment through reconstruction to analysis. Where it kind of creates a package of your RHIT file. Well, now we have a file format called HITS for the raw format, which is actually a closed format. But if you do a reconstruction with these extra APT branches, then the APT file then is also included in this export format. An APT file would be where you would have your all of your raw data that you would like. And then for the analysis section, that is stored in an XML file, and an XML file has can be imported using APT Suite, and will let you see your analysis tree on different machines.
So therefore, for data reporting, you might see this format being the format you might report coming out of an experiment potentially.
Right. Yeah. Yeah. For that, so for FAIR, for instance, I would assume that you'd want to have that entire data package available online. All right. Any more questions?
So I had one. I can't use the Q&A function very easily as the host. Do you have any? Can you comment on whether the custom modules run as fast as the built-in modules, or is there some kind of speed penalty because they're custom code?
They will run as fast as the built-in modules. Yeah. Yeah. I mean, obviously, if you need, you'd have to do your own multi-threading, for instance, if you wanted to take advantage of a large CPU or something. But in terms of access to the data, we do this thing called memory mapping, so that the APT file is memory mapped, and so when you call into the function that you get in your run gives you a pointer to that memory, and you get so because it's memory mapped, it's extremely fast in terms of the I/O of the raw data itself.
That's great. Thank you very much. I think with that we'll possibly need to move on.
All right.
But thank you very much for that. That's excellent. So our next speaker is Jens Guttorn from Aachen. I'm just going to give you remote control, Jens.
Okay. Can everyone hear me?
I can hear you just fine.
Perfect.
Jens, I think you should be good to go.
Okay. Yeah, thanks for the invitation, and I'm really happy to talk about this. It's funny to talk now after this talk about the new features of AP Suite, as a lot of the stuff I'm talking here came from the fact that previously these custom analyses were not possible. So let's see. Yeah, it's working. So previously, IVUS was limited in a way that once you wanted to do something, this was custom, not available from IVUS itself, you couldn't do it, or you needed to wait for it to be implemented, and it might never happen, or it takes a lot of time. And the alternative you had for this was using custom scripts, which for the most part were not really user-friendly, as the moment you share them, it's hard for other people to grasp what you're actually doing. So it can be tedious. So, but with the EPOS file, we could, in theory, get all the data that was collected in IVUS, that was generated in the reconstruction, as well as all the raw data, and get most of the analysis, all the analysis that we wanted to do. So for us, we went to work on MATLAB, as we used the software as a way of generating fast and also fairly complex analysis. It's not the fastest in terms of runtime, but it's really easy and fast to implement new features, as you have all these visualization options already available. So there you have quite an advantage. So in the end, we came up with the goal for our software ePOSA to create an easy to use and also easy expandable platform for expanded atom probe analysis. And the key part here was for us always to create something that's for expanded atom probe analysis, as we were interested in a lot of stuff that is happening to the multiplicity of ions, and this was all not available in IVUS. And we never tried to reproduce or redefine the features that IVUS was actually doing, but just look at the regions that IVUS couldn't handle as well, and try to provide a toolkit that's easy to use there for anybody who might be interested in this. And with this came out ePOSA, as you can see here, which based on the principles that it was fast to install and then easy to use. So you can just install it in MATLAB with a single click, and then load your range file and your EPOS file into the program, and then have a wide variety of data selection options where you can filter your data in terms of ion, in terms of detector position. You could filter in terms of reconstruction position. You can select the multiplicity that you want to use for your analysis, and also you can filter in terms of the voltage, the mass spectrum, and the nulls. So every way that is, every data stream that is input into this analysis is in the end filterable for every analysis that you want to do. This makes it for us then afterwards really easy to implement new ways of visualizing and analyzing your data, as this core principle is included once and all the rest is just built on top of the space, and then can be pushed out if something new is needed within the matter of hours sometimes, as MATLAB provided us with this, so we could easily adapt to new ways we wanted to look at our data. And this is all, of course, available for you. Then also there are nice ways of saving the results, and you can also directly then manipulate them, or not manipulate, but visualize them in MATLAB itself and use the MATLAB features to work with the plots from there. So what options do we have now for data visualization? At the moment, we have options to visualize the reconstructions, not create one, but we can create isosurfaces, with the advantage here that we can create the isosurfaces also in terms of multiplicity, or all the parameters that you saw before. So showcase areas where the multiplicity is higher. You can basically select any value as your isosurface value. We can also visualize detector event histograms, and there's some functions for working with pores, and we can see the evaporation sequence, which ions were evaporated together, if there's some insight for you in this. This came then to our main focus, why we initially developed this, which was the multiplicity analysis, where we could just visualize the multiplicities for different ions and multiplicity in which they occur with which frequency. In addition to this, you could showcase the distance between multiple events on the detector as well in the reconstruction. We can visualize the multiplicity in 3 dimensions, seeing different changes there. There's an option to get correlation histograms out there. You can also filter these correlation histograms and select data part of it there. And we have an option for the co-evaporation analysis that enables you to see which ions evaporate correlated or anti-correlated. And with all this, we already worked a lot with the software over the past few years. Maybe some of you have heard me talking about this 2 years ago in the last Atom Probe workshop. or this workshop here. And we initially started with the quest for investigating crystallizing phase change materials, where we saw really high multiplicity and attributed this to bonding mechanisms. And this was then further extended over the last following years, and even we found we had some nice samples where the multiplicity due to some nanoprecipitates really changed throughout the sample, and all the software that was previously mentioned really guides us in understanding all of this better. And now for a bit of a shift, this was the initial way why we developed this. And then later we had some other things that we needed to solve, which was for us peak solving of overlaps for proximity histograms, where especially at interfaces between two materials, the chances are really high that you have overlaps due to the large number of ions. And there there's no other, we didn't need to know another way to do this then, or to do this without anything that we didn't code. Yeah, so we made our own stuff together with the work of Andrew, which used the natural abundances of the different ions in each bin of the proximity histogram in a maximum likelihood estimation and a maximum likelihood estimation to get some nice data out of the proxygram uncorrected. I could go on for this for quite a while, but as time is limited, I instead just want to show you some case studies that we performed on this. For one, here we have some kappa, indium, gallium, selenide, zinc oxysulfide interfaces, which have many overlaps between zinc sulfur and also zinc oxysulfide with selenium, where when you just run these interfaces, investigate these interfaces, you see that the zinc concentration is underestimated while the sulfur concentration is overestimated. And even if you can correct this, you will still have the problems that your selenium will be overlapping with these peaks as well as on both sides of the interface. So here you can see on the top the before correction and on the bottom below correction, which as we were able to correct this here quite nicely, could correct the zinc sulfur concentration, also the oxygen concentration, and limit all the inferences from all these different overlaps. A second case study was performed on a simulated data set, as you can see here, where we generated 2 nanometers big precipitates in many of them in a small cube, and then looked at our generated composition and the not corrected and corrected composition, where you can see here on the top again, that's especially the aluminium content is highly overestimated. And if you looked on the dashed lines in the bottom, you can then see that the correction is actually working really well. And even though the errors due to this really limited number of ions get a little bit bigger, we still have really high resolution in terms of the proximity histogram itself. There's still a little bit of smearing, but this is mostly just due to the fact that the isosurfaces itself at precipitates that are this small are, as most of you probably know, not perfect and probably can't be 100% perfect, but the solution that we found here is already really nice. So with this coming to a conclusion, maybe for people who want to use these proximity histograms, you just load in your data files. Then you can see here on the right side of the screen that you can just generate your isosurface really quickly in a way by just selecting the data that you want to include. And then you just need to select the range file that includes every ion that's inside your mass spectra. And if an ion is ranged once, the software will find the missing peaks and then calculates the overlaps in this and gives you, with one simple click, the solved proximity histogram. But overall, we created thus a user-friendly and expandable, fast expandable tool set, which with Etiprop Suite 6 might not be so needed, but for now was quite nice. That has in the key features these easy data selection options, and then a whole bunch of options for analyzing multiple events and also generating proximity histograms with solved overlaps. And if you're interested in all this, and would like to try it, just write me an email. This is most of this is developed by myself, and there might be some errors. So I like to stay in touch with people who are using it, that I can help them when they find something that's not working as intended. Yeah, thank you.
Okay, thank you, Jens. So I guess this sort of hinted at the end of your question there. So how do you think that users are supposed to interact with that? Are you going to be providing a link ultimately to the software, or is it just for now it's email? How's this going to work in the future?
So in the future, maybe I'll provide a link, but sometimes, especially as MATLAB versions for everyone who's using this sometimes change. I like to stay in touch with everyone who's working with it, but just write me an email. I will provide you the software itself. You can just install it in MATLAB then itself, and inside the software is something that I included that when it starts, it checks for a new version and automatically updates it. So even if we don't stay in touch, we'll always get the newest version. Besides this, I just like to know who's using it for the moment, and maybe I'll publish it, but I'm not sure yet.
Okay, so I guess another question. I mean, for people who aren't, for other laboratories like the Stuttgart laboratories or other people who are building their own equipment, how would you see them interacting with this software?
If they want to interact, you just need the range file and the EPOS file. I saw for the people from Stuttgart that they can export EPOS files. So if they want to just export them, or if they just export their files, they can then quickly use them to import them into this software and use it. So
okay.
Yeah,
we've got another question about proxygrams. In terms of do you have enough statistics to use the natural abundance in each of the proxygrams?
Yeah, it depends, of course, on the interface you are looking at. We are currently on a way we submitted. So Andrew and I submitted a paper about this, where everything about this is discussed in more detail. But if you have a proxygram, it depends on how big your interface actually is. And you can, of course, then increase the number of ions that are included by just increasing the bin size. And depending on which interfaces, you might then lose a little bit of resolution, but probably most interfaces that occur in AtomProbe should be investigatable with this solution.
I think we've got time for one more question, just in the last minute. So I guess the question we've got is for people who can't generate EPOS files or don't have the capability, how might they go about interacting with your software directly if they don't have access to IVAS?
If they can't generate EPOS files, I think they just would need to contact me. I think it's quite quick to, once they have the data, to create a new script that just imports different file formats. Up to now, it wasn't really necessary, so there's nothing in there yet, but I'm happy to provide something for people who would need it.
Okay, I think with that, we can move on to our next section. Thank you very much, Jens. That's great.
You're welcome.
So the next section of this talk or meeting, sorry, is inspired by what happened 2 years ago at APTNM 2018. There were a series of flash talks. So these are designed to be short talks to introduce a particular topic, script, software, solution, with some short Q&A afterwards. So with these, again, quite important that we keep fairly to time. I'm aware this is a long meeting. After the first 4, we're going to have a very short break. So there will be light at the end of the tunnel, and hopefully we can stay refreshed and ready to ask lots of interesting questions. The first one we have as a recording. Benjamin, I believe, is on holiday, maybe with some patchy internet, so we have him on the call, but we're going to play the video for this one, and then we can go to questions live. So let's go.
Hello, my name is Benjamin Kaplans. I'm a researcher at the National Institute of Standards and Technology in Boulder, Colorado. And today I'll talk to you about a method we're developing for the voltage and bowl correction of delayed evaporation in multi-hit events. So one of the goals we have at NIST is to extract as much information as possible from these atom probe data sets. So basically let no ion be uncounted if it doesn't need to be. And so one way of extracting more information is to look at these correlation histograms. So here's some work from Karen Kruska and Dan Schreiber, where they noted that the red dots in this correlation histogram are the events or the ions that are typically counted and ranged. The green dots are the ones that are typically just background. However, there's quite a bit of structure here, and where there's structure, there's information. So with some help from David Saxe's work, they noted that you can replot these in a histogram across delta time of flight space. You get out these nice sharp peaks, and you can actually recover the majority of this background. You can assign them, and you can see how that might bias a composition measurement. So we have some data which was taken at relatively high fields, so there's a decent amount of background, and also with very long periods for the laser, so low repetition rates. And so we think that this DC evaporation is worth investigating, and so we'd like to kind of do this type of analysis. And so naturally, when we went to do it, it didn't work that well. So basically in this lower left quadrant of this time correlation histogram, what you see is a lot of cross peaks, as you'd expect, and you also see these diagonal lines, which are the events that are correlated to each other, but uncorrelated to the laser pulse. Up at this upper right quadrant, what you see, unfortunately, is instead of these sharp lines, which we might be able to interpret, we see these diffuse bands that contain very little to no information. So the question is, this is the region we actually want to be analyzing for the purposes of this work, and so why are these bands? What's happening here? So what we eventually figured out was it's an issue of time zero. So time zero is simply not well defined for these events that are uncorrelated to the laser. And so the reason that's a problem is because the voltage and Bowl correction need this T0 corrected time. And so what happens when you use the wrong time is you actually uncorrect the data, you deteriorate the data. So for example, at 5 microseconds, you're applying roughly 10 times more voltage and Bowl than you should be. And so the question is, can we do better? So a first order way of trying to fix this, so a voltage and Bowl correction for these laser uncorrelated multi-hit events, we approached as follows. We determined the voltage and Bowl corrections from the single hit data, or the 1D histogram data. And then we use the time of flight. We actually subtract off this number that's given by a function delta. And this is a function of the flight times for a set of multi-hit events. And so then we add that back on once the voltage and Bowl corrections are completed, because that way we don't overcorrect. And so this function delta is really just an estimate for T0. And the simplest estimate we could come up with that seemed to be reasonable was simply take the mean time of a set of multi-hit events and then subtract off an empirically determined constant. So how does this actually work in practice? It actually works surprisingly well. So if you now look up at this upper right region of this correlation histogram, you can see all those broad diffuse bands are now very sharp lines. Kind of interestingly, if you look at this lower left quadrant, where you'd expect to see very sharp cross peaks, those have now turned into somewhat diffuse peaks. And if you think about it for a little bit, you realize that makes sense, because instead of using the time zero that was very accurate for these laser correlated events, we threw that out the door and ended up using this very rough, crude approximation delta. So some final thoughts are that the naive assumption for T0 is not correct for these events that are uncorrelated to the laser pulse. And this means that the voltage and Bowl corrections actually deteriorate the uncorrelated data. A first order correction works surprisingly well. Determining this function delta may actually be a good application of machine learning methods. Right now it has a single free parameter, but the data may support quite a number of free parameters and may improve the correction a bit. These type of corrections really need the rawest form of the data possible, and they need to be in portable data file formats. And furthermore, this work was all done in Python because, to my knowledge, existing software packages don't let you explore these type of things very easily. And future work will go into developing and identifying. We'll go up into identifying the peaks in this delta T histogram and trying to quantify and extrapolate those to the full time between laser pulses, to see how that may affect composition. So thank you for listening. Thanks to the funding sources, and please feel free to contact me at benjamin.kaplans@nist.gov if you have any questions or suggestions. I'd be happy to hear them. Thank you.
Okay, thank you very much to Ben. Ben, do we have you on the line?
Hopefully. Can you hear me?
Excellent. I can hear you. Yes, I can. So we've got a question, whether or not have you looked at something like doing Huff transforms in order to arrange correlated events? Is that something you've been considering?
That's not something I've looked at, and I wouldn't actually understand exactly how to use it. So I'd be happy to talk some more to understand how to use a Huff in that context.
In terms of the T0 information, you're extracting that from the sort of the other ions within the spectrum rather than just the multi-hit ions, if I've understood you correctly. So in terms of, do you think it's possible to gain both this extra information that you're getting that allows you to sharpen these additional broad peaks as well as to maintain the original mass spectrum? Because it looks like there's a trade-off, one or the other, but it's not clear if you can get both.
Yeah, so that's, in theory, it should be possible. So basically, if you were to look at that, if you rotate those correlation histograms 45 degrees and remap them, then what you would see is at a cross peak, everything is very sharp because T0 is exact. And as you move away from T0, both to negative and positive time, things get broad. So if you appropriately parameterize that two-dimensional space, you should be able to put a lot of the information back together. From a pragmatic perspective, I was interested in 1 microsecond and onward. So if I mess up the early times, I can just treat those as 2 separate cases.
Okay, so I guess we've got a question. Someone's asking whether or not this is actually simulated data. I think you're probably best positioned to answer that one.
It is all very real data with all the problems associated with it.
Excellent. Yeah, no, it's great to see that you can actually get these statistical improvements out of the data. So how do you see this going forwards in terms of making this kind of code available or these kinds of analyses available to the community?
So at present, it's just under research development state. Very broadly, the things you need to make this type of thing work is you need voltage and bowl corrections that you can do yourself. And so, those are things we've kind of worked up in-house. They're not perfect by any stretch, but eventually we like to share those.
Hello.
But anybody's welcome to contact me for code, and hopefully eventually it'll be available online. We have some previous code in a similar flavor of work that was published on GitHub recently, but this is not yet ready for that.
Okay, and I think there was just one comment from a user. They're sort of maybe thinking about some work. Apparently, Yimeng Cheng may have done some work regarding that as well. So it'd be interesting to see if you think there's any additional information you guys can pull out together. I'm not exactly certain what the questioner is implying in terms of how you could combine those 2 efforts, but I think it would be worth having a bit of a chat with that user. All right, thank you.
Sounds fantastic. Thank you.
All right, thank you very much for that. And Andrew, I think you're up for our next talk on R for atom probe tomography.
Okay, so just want to give a brief introduction on this first. What kind of motivating why we were using R to develop this package. So R is an open source kind of language that is really used by the data analysis and statistics community, and provides a wide variety of packages and modules to perform particular analyses. It's really been developed in order to have kind of rapid data analysis and visualization techniques, including things like interactive plots, 3D plots, et cetera. And all those packages are maintained by subject matter experts. And so it means that the burden of getting those analyses and maintaining those analyses and ensuring that they're correct is distributed throughout a wide community. So we developed our particular package to kind of add APT analysis functionality using R and take advantage of all these packages to make an easily extensible open source analysis suite. Now, I will admit right up front that we're continuing to work on this, so it's very much in its alpha stages. Things work, but are continually being added and tweaked in order to extend the functionality of individual routines or add new routines. So far, we have features that a lot of people have shown so far, like correlation histograms, detector analysis, mass spectral fitting. And kind of the main focus that I'm going to talk about today is leveraging the formal spatial statistical tests that we can use on three-dimensional data to do things like cluster detection and characterization. So kind of the resources that we use to build this are R, which is available kind of online very easily, this GitHub repository that I linked to here, and then for my personal use is kind of just a helper thing is this IDE called RStudio, which is kind of the de facto standard for the R community in terms of development. I will be sharing kind of code snippets embedded on the pages. For the most part, those are complete. Once we get into kind of the machine learning process, I've just highlighted the kind of main workhorse functions there because the full code would be too much to include on a slide here. So the first thing we need to do to use this package is have our APT files. So here's some examples of basically we can load in just by pointing to that file. We can edit that range file so we have more custom names that kind of differentiate the charge states or particular isotopes in that range. We can do kind of the basic things like plotting out our mass spectrum in kind of a very easy way, utilizing a package that's designed for mass spectral analysis. So it has a number of built-in peak finding algorithms, background correction, et cetera, that once again are really specific to the mass spec community and are maintained by them. So once again, leveraging that development. We can then take those mass spectra, generate these correlation histograms very easily, and even look at higher indices by basically taking the multiple hit data that exists and looking directly at what those hits are and look at kind of higher dimensional spaces than we could plot in this kind of 2D correlation histogram. We can also look at kind of the detector space that's available and look at things like charge state ratios. Once again, we've kind of adjusted that range file slightly to mark which peaks are in which charge state, so we can look at how those things vary over detector space or in three-dimensional space as well. And that's kind of where we're really focused our development, is working on analysis techniques for three-dimensional space. So here's kind of a basic, this same data set where we can just visualize what these different types of molecules look like and where they reside in space. The analysis that we're going to be doing is really leveraging this SPATStat package, which is developed by the experts within the spatial statistics community to enable all kinds of statistically rigorous analysis routines in kind of spatial point pattern data, which is what we have in AtomProbe. So to walk through an example of this, I'm going to be looking at this kind of cluster data set, which are kind of alloy molecules within an aluminum alloy, and kind of try to characterize these clusters using these spatial statistics methods. So we can load in this data, plot this data, kind of all through a few lines of calls here, and then start looking at what the kind of aggregate properties are of this data set when we're looking for clusters and trying to figure out what is actually going on. So I'll briefly breeze through in spatial statistics. If we have clustering, there are a number of statistical functions that we can do to summarize what's going on here. This is one in particular. I'm not going to go into it, but essentially we can look, test for clustering, and extract features from this function that we can then feed into a machine learning program, and that can tell us information about what those clusters actually look like. This work is in a paper that we submitted, and if you're interested in that manuscript, I'd be happy to pass that along offline here. But when we do this, we get kind of results that allow us to estimate the radius of those clusters, the density of those clusters, and kind of the background density of those clustering type points. We can estimate the errors on this based upon kind of our training and test data. So we know that when we state a figure, what our level of confidence is in that figure. Similarly, with that information, we can then do prediction on our observed data, where we simulate a pattern that uses all these parameters that we've tested in our trained machine learning algorithm, and do prediction and compare that with the spatial statistics function. So if we compare that simulated data with our original observed atom probe data, we can see that for the most part, we match the distribution of those points, and that gives us a pretty good idea of where those things are sitting. So all of this basically lets us kind of characterize aggregate properties of clustering and identify those features. So I'll just kind of summarize here is you can look at this package, you can find more tutorials and examples of other things you can do, including interactive plots at this website here. If you're interested more about spatial statistics, this is a really useful reference that is developed to be used in R, and some other R references here. Feel free to follow up via email, because I know these are going to be archived. The email listed there is not my own, but our group leader, who will kind of be a more permanent email address. And with that, I'm happy to take any questions. Thank you.
Okay, thanks very much. Just remember to raise your questions, like audience, if we have some. So I guess the machine learning is not something that's really been explored very much in our community. There's been a couple of small shots at it. What do you think are the sort of key areas in machine learning that we need to be focusing on to improve things like clustering analysis?
Yeah, so I think we really need to be taking advantage of all the data that exists in our Atom Probe data sets. So far, we've kind of been segmented into either mass spectral processing or spatial data processing or multiple hit processing. And I think there's a really rich data environment that allows us to combine those elements together and discover kind of these higher order correlations between all these different things. And that's kind of what we're starting to do here with this clustering analysis set, where we have much more formal and rigorous tools to test for clustering. And then the next level of this, instead of using a range file, which is what we did now to kind of convert these continuous marks of mass spectral, charge to mass ratio into particular identified species, is these data analysis tools actually can take advantage of the raw mass to charge ratio associated with each point, and then leverage that to do better kind of spatial segregation of peak overlaps using their spatial position, and starting to be able to get better identifications there. And really, one of our long-term goals is being able to not only look for clusters, but really identify and have a classifier of morphology in these systems, where we can take a data set and say, okay, are we actually getting precipitate type information, or we have something more like a bicontinuous network? Our group really focuses on kind of small molecule organic systems, which have pretty different characteristics and kind of features of interest compared to a lot of the Atom Probe community. And so I'm really keen on getting feedback from this group and more broadly from the community about what features are really valuable to extend these analyses in a way that is broadly applicable.
That's great.
Okay,
I'm just going to jump in and say thank you very much for that. I'm very much looking forward to having a look through your online tutorials later. Our next presentation is being presented by someone who unfortunately can't join us live, but please do post your questions in the chat, and we will forward those on, and we'll make sure that there is a response sent out after the meeting. And just to remind you that a detailed list of all the software packages and links will be made available after the meeting, as well as the recording to the meeting as well, so you can catch any details you missed again.
Hello, and welcome everybody to my talk on heap data treatment in R and Python. My name is Sebastian König. I'm working at Polytechnique Montreal in Canada. And as the title indicates, I'll give you a brief overview of the kind of scripts I've used in R and Python and that I wrote over the last 10 years. I won't be able to answer questions, so if you have questions, please tell them to the organizers or write me an email with the email address that you can see down there. So as already indicated by the title, there have been two software packages I've mainly used for more advanced data treatment. One is R and one is Python. I'll briefly introduce the packages and tell you what I've done over the last few years. So R is basically a programming language similar to what you would have from Python or Java, but it's developed for statistical data treatment and data mining. And if you install just the language, you just get a console, and you install RStudio with it, you get something like a MATLAB Lite, which is significantly less powerful than MATLAB, but has two advantages. A, it's free, and B, the code is extremely easy to maintain. So you can.
Stopped again.
All right.
Programming language similar to what.
Sorry about that, everybody. Just trying to get the right place.
Data treatment and data mining. And if you install just the language, you just get a console, and you install RStudio with it, you get something like a MATLAB Lite, which is significantly less powerful than MATLAB, but has two advantages. A, it's free, and B, the code is extremely easy to maintain. So you can use the same code for tens of years on multiple platforms. So once you've written code, you can basically use it forever. So the scripts I've written for this language are a whole bunch of scripts to read and write data. So I have scripts that load and write POS and EPOS files from IVAS, as well as load the range files that we typically use. I have scripts that write out PLY files for Blender, which you can load in as a point cloud in Blender, so you can see the point cloud in Blender. I have scripts for Aviso, which write AM files. It's basically can be used for cloud rendering. I'll come back to this later. Can read and write the TAPSIM files, the bin files that allow you to check atom position and trajectories and everything in TAPSIM. And I can read files from LAM-MPS, the dump read, so you can check how the atoms move in the Monte Carlo simulations. The molecular dynamics simulations in LAM-MPS. In addition, I can do 1D profiling on IVAS data, or basically on any of those data, both based on elements and based on ions. I can do it based on an axis and based on a circular profile, basically making successive cylinders or successive cylinder tubes to create circular profiles. I have a simple background algorithm for all of these, which basically just tries to remove the constant time background. And I can do this, so the axis can be based on an absolute length like nanometer, or just counts tens of thousands of ions or something like this. I have 2D mapping scripts that based again on for elements and ions, with and without the background correction again, and you can do it based on the coordinates from the reconstruct, or just based on the detector coordinates, the mapping that the two-dimensional mapping that you would do. Then finally, 3D mapping. So similar to what IVAS does, I can calculate the Gaussian, can calculate on a grid what happens if you would spread the atoms with the Gaussians, and export this to this grid then to Aviso, and that is where you get these AM files, and you can make the cloud renderings that would give you like over color map which concentration of which element is where in this three-dimensional volume. And finally, I have some scripts to generate lattices. So this is basically to make test data. And since I'm mainly working in semiconductors, I have it for diamond structures, zinc blende, lenstelite, and wurtzite, which are the cubic and hexagonal structures for like silicon and gallium arsenide. So this is what I have for R. With Python, I've only been starting relatively recently. I'll have to remove this a bit. And so I've only used it basically for the last few months intensely. And so on the right side, you see all the libraries I've been using already. So aside from Python, I've been using SciPy, NumPy, Pandas, and Matplotlib to organize the data and use some of the functions that those provide. And so far, I've written scripts that again use read and write POS and EPOS files and read the range files from IVAS. I can do 1D profiling along only one axis right now, basically only along the z-axis and only for elements. And right now only based on lengths, but of course changing this would be relatively straightforward. I can do lattice generation again, where I can make right now simple cubic and diamond lattices But again, changing this is not too difficult. And so the main points I'm using this right now, it's because it has some more powerful algorithms than R. So I try to do mass spectrum peak identification. So right now I can find the peaks, but I cannot associate them with anything. And I do Voronoi tessellation of the two-dimensional data we have. And so for right now, I can do the Voronoi tessellation and I can make cuts through them and plot the cuts. So something like this, for silicon germanium would be a cut through an atom probe measurement, which the tessellation for each of the type of atoms. And that's basically all I have so far. If you're interested in any of this, again, let me know. You can write me an email. Ask the organizers are happy to help you out if you need any of these or want to use any of these scripts. Thank you very much and enjoy the virtual meeting.
Okay, great. Well, thank you, Sebastian, for that. That was very interesting things that you've been working on. I'm looking forward to contacting you. If any of you have any questions, please feel free to post them in the chat. And like I said, they'll be forwarded on and hopefully we'll get some discussion going and you'll get answers to your questions. Our next presenter is from the University of Oxford, and Phil is going to be, when my PowerPoint works, Phil is going to be talking to us about sulfur isotopes. So we have him on the call, but he's provided a video. So Phil, if you could stick around and answer questions at the end, that'd be great. Thank you.
All right, I'm Phil, and we'll be talking about sulfur isotopes with the Atom Probe in the script that we wrote to do that. Mostly I'll be talking about pyrite, which is an important mineral in ore deposits. We use the sulfur isotopes to determine the source of the ore fluids, among other things. The problem analytically is that it's a very complicated mass spectrum. Lots of different elements are present here beyond just Fe and S. There's lots of molecular peaks as well, and we're looking for really small differences in the sulfur isotopes. This is the sample we were trying to do this with in the first place. It's from Nevada. It's a gold deposit, and the gold is actually hosted in this arsenic-rich overgrowth on the arsenic-poor pyrite core here. You can see that's really fine oscillatory zoning. We wanted to see the difference between, number one, the core and the rim in terms of the sulfur isotopes, but really if there's any differences between all of these different growth zones in these sulfur isotopes. This was something that was done about 10 years ago with the NanoSims, and so they found relative differences, but they weren't able to do this quantitatively. Again, we're looking at the third and the fourth decimal place, so it's analytically quite difficult. For us, it's an overlap problem. Every place that we try to look at a sulfur peak family, there's some sort of an overlap. Most of these problems we can't actually solve. On the main 32 to 36 peaks, there's the double sulfur which overlaps, which is just too difficult to solve. At the S2 peaks, though, we have a shot at it. The problem then becomes there's 4 sulfur isotopes, and there's different combinations of those isotopes that can give us the peaks between 64 and 70 Daltons. So how do we go about this? So the first thing that we do is we use Jesse's code to very systematically determine the area under the peaks and get a single value of counts at the 64 through 68 peaks. Then we use those numbers and we start making guesses. We tried doing linearly least squared. We tried a couple different ways to solve this overlap problem, but in the end we settled on this sort of brute force approach, which is really just we make a guess, we start with the natural abundance, and we just populate 2 rows with the abundances of those guesses. So literally just the number 32, 33, 34, and 36 in row 1 and row 2, the same isotopic numbers in row 1 and row 2, and we randomly pull from row 1 and row 2, we combine it into row 3, which simulates the random combination of the atoms as they're coming off the tip to form our molecules. So we then have a simulated mass spectrum in row 3 here, and we can compare that to the real mass spectrum. We can calculate a mismatch number for that guess, and then we go to town. So we use the natural abundance as sort of the starting point, but we go left, right, up, down, every single way we can think of in terms of iterating the 32, 33, 34, 36 sulfur isotopes to we start guessing and guessing and guessing and doing a grid search of guesses. At each grid point, we have a mismatch, and we have then a 4D sulfur isotope space. Here everything's plotted to 34, so we can actually show it on a 3D plot. And then we end up having the red is more mismatch and the blue is less mismatch. So the intersection of these mismatch planes is our best guess, which is what our code spits out. And then we can actually use our code on real data. So we started off with some simulated data from Andy, where he started with a known number of sulfur isotopes, and we were able to match that quite closely. We used similar samples to what the NanoSIMS study looked at, and we saw the same heavy, light relative differences. We published a paper on that, which is really nice. We also could look at the arsenic peaks, which the ASS peaks are shifted to a clean part of the mass spectrum, and we can compare to see if the double S peaks gives us the same answer as the ASS peaks. And not all of our data sets have arsenic, so to be consistent, we use the double S peak for everything. After that, we really went to town with some reference materials, and we ran them all sorts of ways that we could think of on all the instruments that we have available at Oxford. And then we, in the end, were able to get a fairly good deviation from the nominal values, which are in the top row there. This comes with a big caveat, is that we had to do a lot of scratching our heads in terms of the instrumental artifacts. So there's a very specific set of conditions that gives us the right answer, 80 picojoules. We've only managed to get it to work on the XR. There's a very specific voltage range over which this works. And how we define the grid search also seems to have a bit of an influence on the numbers that are put out, sort of whether it's an even or an odd number of bins and whether we end up on a local minimum there. The first paper on that is what came out last year, which is presenting the code, but only really for the relative differences. We've done a lot more work now, and hopefully the next paper will be out this year, hopefully, where we'll also release the code then with that. It'll probably be in microscopy and microanalysis. Thank you for your time.
All right. Thank you, Phil.
All right, I'm Phil.
Oh, Phil's again. Do we have some questions for Phil? I have a question. Are you on the line? Can you hear us?
Here. I can
Excellent.
hear you.
I have a question about your running conditions. Do you think that the running conditions you've chosen are correct, or is it just that two wrongs make a right?
Well, our run conditions were we tried everything that we could think of, and where we got the right answer with the reference materials, that's where we settled on.
Okay.
You know, it's not really the right way of doing it, but we were able to reproduce the analyses a number of times. So we're pretty confident that for some reason these conditions seem to work. Not 100% sure yet why. It probably has to do something with multi-hits. And we're still scratching our heads about that. But at the moment, you know, we seem to be able to get the right answer. We seem to be able to do this consistently, and that's really what matters for us.
Great. There's a question from Dan. I think this is with his software development hat on. What do you think the main sort of areas for software development are for people working on geological specimens?
I mean, for us, the big thing is being able to look at chemistry very carefully, and isotopes are something that geologists are very interested in, looking at isotopes on the nanoscale. This is something that, you know, the guys at Curtin have done. This is our foray into this using sulfur isotopes, and very specific systems seem to work and others don't. And I don't want to talk about the lead stuff. That's Steve Reddy's and the other guy's domain. But yeah, I think isotopes and being able to do something proper with that is really where it's at.
Okay, great. We have a question. What is the minimum amount of sulfur required in the material for the approach that you presented to work? Would it also work for sulfur impurities in alloys, or is that too low of a concentration?
That's a great question. So at the moment, we've only really tested it with pyrite. We're not even looking at other sulfides. So FeS2, so that's 66 atomic percent sulfur. That's where we're comfortable with it. Mm-hmm. As soon as you start getting other elemental impurities, we start worrying about other overlaps maybe. Yeah, for now it's a very niche. It's a very niche thing. Pyrite is a very important mineral, which is why we're doing it. But yeah, I wouldn't be that comfortable putting it outside of this little comfort zone yet.
Okay. I think we do have a little bit of time. We have a break, but I'm gonna be cheeky and ask another question. Put you on the spot while we've got you. How many multiple hits? What detected multiple hit fraction did you have in these analyses?
I can't remember off the top of my head now. It wasn't that small of a number though. We're working a little bit with Kamika. TyPros was starting to do some work for us before COVID. So we haven't managed to get any analyses done yet at Kamika with their instruments, but the multi-hit is something that they're looking at more than we are.
Okay.
Again.
Great. So it's always something I'm interested in. So just briefly to mention, you mentioned Jesse's peak fitting. I thought it'd be worthwhile just to explain for people. This is a slide that he sent me. He's using a MATLAB toolbox that was, I think, originally developed for X-ray peak analysis, but it's, I think, a general peak fitting tool. And he's been using this to quantify the counts from peaks. And so I believe Phil's been using this code. So just to put that up there and mention that that's what they've been doing. Okay, so we've been going for a little while. I think it'd be great if we all just take a 5-minute break. If we come back at 22 the hour, whatever hour it is, wherever you are located, I suggest you take what gamers call a short bio break for whatever refreshments are required. Stand up, have a stretch, look out of a window, whatever it is that you need to just step away from the PC for a moment and come back in about 8 minutes time. I will be back here slightly before that. And then we'll get on with the next half of the Flash Talks, the technical committee discussion and Q&A at the end. And I will be the next Flash Talk. So yes, make sure you're back in time for that.
Nobody's got the camera on. Everybody's trying to hide, right?
Yeah. Let's see. Stretching silently and privately. Got some delay on the YouTube video. Almost 10 minutes.
Okay, so we'll be starting back in just a moment. If any of the panelists had any discussion points they wanted to raise quickly before we leap into libatomprobe. So I'm looking at my second screen and everyone is arrayed horizontally across the top. So that's why I'm always looking over to the right. Is that better? Maybe. Peter, I take it you're all ready to go for your live demo after Marcus?
Yes, I will mostly be showing what we can do. I won't actually run a lot of code because I ran everything before so that I don't run out of time.
Yeah, no, that's great. That's good. It's organized.
Nobody likes the sound of error messages because I mistyped something.
All right, I think we should get back underway. We still have pretty much all the people, so hopefully we've not lost too many, and those that are apparently here are actually here and listening. So welcome back from our break. I'm now going to put my libatomprobe hat on and talk to you about the C++ library that Dan Haley has developed and that I'm also a co-author on. So what is libatomprobe? It's an open source library for general purpose atom probe computation. So what does that mean? It means we have lots of different algorithms, and these are all standard. We only implement things which are published. Different algorithms for doing, say, statistics, for doing file input and output, peak overlap solving, various different things. And the idea here is that with a fairly permissive open source license, you can take this library and use it in as a fundamental building block in your code and applications. So we want the code to be robust. Most of our functions are covered by our unit testing, automated testing, and this maximizes the reliability and accuracy of the code, so you know that it's correct. We try to parallelize things as much as possible on the local PC so that you get the best performance from the code, and it's written in C++, so it's nice and fast, and it's documented. I'll mention that at the end. So the idea is that this is aimed at people with more coding knowledge than just say average PC user, but a lot of scientists develop their own code, and we're looking to optimize and take away some of the the tedium and the difficulty in reinventing the wheel that happens when people re-implement common functions. For example, anyone who's written a RRNG range file reader will know the headaches that you can have. So we've got code in a standard library that you can use. It compiles on Linux, Mac, and Windows. On Windows, we're using either Cygwin or the Windows subsystem for Windows subsystem for Linux. You can clone the repository. It's fairly straightforward to compile. I'm not an expert at actually compiling things, and I found it relatively straightforward. You can also use it with Python. So you can use all of the power of the AtomProbe library and basically just plug that into Python and use it interactively rather than having to compile C++ code. Currently, this is mainly Dan Haley's work, also mine, but we're looking for anyone who wants to contribute, and we'd really like to try and make this a part of a lot more sort of general purpose AtomProbe software as broadly as possible. So it implements a lot of existing algorithms, like I said, parallelized and optimized code. This doesn't stop you from using your own code and doing your own analysis. So ideally, you wouldn't even know really you were using libatomprobe. It would just be something you can use on the background in the background. Whenever you need to open a new file format that's come out, we can implement that, and you can just call open file or whatever, and then libatomprobe would handle that and spit out the same formatted data that you would be expecting opening a POS file or an ePOS file. It's open source, so you can always see exactly what's going on, and you can check the consistency of the code and look for bugs and check exactly how the algorithm is working. It's C++, so it's very portable. There's no need for, say, a specific graphics card to run some particular algorithm. Any desktop or laptop will do. And we'd be very, like I said, we'd be very welcome for anyone to contribute, especially if you just want to add in a small module that's very simple to add in, and that's how I started contributing to this project. So we'd be really keen to have people do that. We've got a little live demo of how this works in Python. So we just simply import AtomProbe, and now we've loaded up the library. We can make a vector of ion hits. This is how we store the data. You see, initially it's got 0 length. Now, if we load some random file that doesn't exist, we get an error code, and we can decode that to get the fact that this file doesn't exist. We could open a real file and load some ions. And we can set an output vector of ions. And what we're going to do now is just loop over all the ions that we've loaded and crop those based on their X position. So if they have X greater than 0, then we're going to add them to our output vector. And now we're just going to save that vector of ion hits as a TAPSIN binary file. So it's a very straightforward example of showing how you can use the library and how you can say, do, in this case, quite simple operations, but you have the power of all of the data and any of the operations that you'd like to perform. So just to give you a comparison of how it would look in C++ or Python, in C++, you would write C++ code. This could be part of a larger application or just a small, simple kind of app that you want to write. It doesn't have to be very big. And the blue code here between Python and C++ are the library-specific things. So that's what you need to know from the library in order to use it. In Python, like we showed, it's very straightforward. You just import AtomProbe, then you use these language-specific bindings in Python, and this gives you Python-typed variables, which you can use however you want in native Python or with any, with pandas or NumPy, whatever you want to do with that. So we enable you to get the data. You can use the library functions if you wanted to do spatial analysis or even reconstruction or whatever it was you wanted to do. But then you've also got all of the power of Python to execute as well and leverage. And all of this is documented with our sort of automatically generated Doxygen files, which I'm showing on the right-hand side here. It's easy to search through. This is automatically generated from the code, so we know that it's up to date. We have various different examples, 15 C++ examples and 10 Python examples, including proxygrams, isosurfaces, correlation histograms, overlap solving. So there's lots of example code that you can just copy and paste, modify as much as you need. And previously we've done online tutorials, and it would be great if there was enough demand for us to do that again. So in summary, we're looking to expand the algorithms that are available. For example, integrating this better with 3Depict so it can use libatomprobe directly. We'd like to support more languages, for example, R, and I think we've seen there's quite a lot of momentum for R, or Octave or MATLAB. It's possible to do that. And if there's demand, then we'd like to do that. For example, I'm looking to merge the code from AtomProbe Lab, which is my own MATLAB code, into libatomprobe, specifically say that peak overlap solving and maximum likelihood estimation, that's being ported into libatomprobe. So that's kind of hopefully going to give that code a new lease of life and allow it to be used more widely. And yeah, with that, I'd like to welcome any questions. I will now look over at the questions and see what questions there are.
Yeah, thank you, Andy, for this great talk and initiative, and also update on your two recent work. There are at the moment no questions in the chat. So are there specific questions to the tools that Daniel and Andy have been put forward and explained?
Everyone's still on their break.
Yeah, I have a question.
Yeah, Peter, go
ahead. So Andy, obviously, so as far as I've seen, everything's command line. So what do you do with cases where user interaction, graphical user interaction is needed?
So libatomprobe is meant to be that kind of basic building block that you can call in a larger application. So we're not imagining really that someone would use libatomprobe directly. So the Python example shows how you could integrate libatomprobe use in a larger Python application that you were working on, or as part of your Python toolchain. I've used it, for example, where I've written my own interface, but the kind of back-end computation is all done using libatomprobe. 3Dpict is Dan Haley's atomprobe visualization software. That's using a lot of common code, even if it's not using libatomprobe directly. So in the future, I think Dan's plan is to try and integrate those closer, so it is directly dependent on libatomprobe.
Okay, there's a question from Deodata Schinde. Do you have recorded videos for the online tutorial you have conducted in the past?
That is a good question. I will have to defer to Dan for the answer to that.
Thanks, Andy. Unfortunately, we don't actually have a recorded video of that one. We did attempt to record it, but unfortunately the video ended up being corrupted at the end of that, and I didn't notice until the end. But if there's specific features or modules that people want us to do some pre-recorded videos, we can do this and make them available. Really, because it's quite broad, the sort of how do we focus and narrow down exactly what the community needs is an open question for us.
Yeah, exactly. Okay, I think with that, Marcus, we should move on to
ask a quick question, Andy. It's Tim from Kamika.
Go ahead, Tim.
How are you licensing libatomprobe? Is it something that we could use and distribute with
right, so
atomprobe suite?
Yeah, so it's a GPL3 license, which means you're not allowed to sell it, but otherwise it's pretty permissible. So I think that you'd be able to use use it as part of a third-party add-on that was available for people to download and use.
Specifically, it'd be helpful, and especially if you had a lot of various file format support, maybe it'd be the best way for us to include support for all sorts of other people's formats.
Yeah, that's a good idea.
Okay, thank you.
Okay, sorry.
To add on to Tim's question, so I think there's nothing that's stopping users from being able to directly access that as part of their, I guess, AP Suite 6 call. There might be some technical things that need to be tested just to make sure that all works smoothly. But I think distributing it as a side thing that you don't directly link to, there would be no issues with that.
Okay, so then we should move forward to Charles Fletcher. Charles Fletcher is a PhD candidate in Oxford, also working with Daniel and Michael Moody. And his talk will be on Castra, a tool for 3D modeling of future operation. And
cool. Thanks for the thanks for the introduction, Marcus. My internet's being a little bit patchy, but hopefully it won't cut out. So yeah, today I'm here to talk about Castra, which is a simulation tool I've been developing during my PhD. Let me see if I can actually go to the next slide. Oh, there we go. Okay. So yeah, Castra is a tool for simulating specimen evaporation in full 3D during atom probe experiments. And these simulations, because they're basically modeling the tip as a continuum rather than at an atomic level, these simulations are quite fast. They're under 20 minutes on a sort of standard laptop. And basically, another additional factor of these simulations being so fast is that they can be used to actually directly drive the atom probe reconstruction, which I'll talk about in a bit. So there's just an example of the simulation of a low field precipitate and its subsequent reconstruction. You can see the sort of narrowing distortion that occurs. So a quick breakdown of what Castra does. So the sort of first step in the sort of procedure would be to set up the model geometry. And this can be done either through analytical functions that are provided, part of a module, or actually you can sort of load in a tip geometry, an experimental geometry using electron tomography. And then you go ahead and simulate the specimen evaporation using Casra. And then following this, you sort of can calculate the ion trajectories. And then basically, if once a termination conditions reached, such as the size of the specimen, it'll either, if this hasn't been reached, it will iterate and continue evaporating the specimen. And if it has, then it will output data. And this output data is in the form of a VTK format, which is a sort of standard computer graphics format, and POS files as well for the sort of simulated reconstructed data. And then a number of additional tools are also provided in the package, one of which is to perform this model-driven reconstruction. The other is to just perform standard point projection reconstructions on simulated data. And then finally, there's some tools to calculate composition and density throughout your reconstruction. So the first thing that you can use Casra for, which is pretty straightforward, is that just sort of some simple comparative modeling with experiment. So basically, yeah, the idea is you can use Casra to understand physical distortions that are observed in experimental data. So by setting up your model geometry to resemble microstructure you might be seeing in experiment, you can basically generate synthetic data of the APT experiment, and then reconstruct it and sort of compare the distortions that are occurring with the experiment to sort of better understand where the distortions are coming from. And the additional feature, this model-driven reconstruction, so basically Casra models the specimen evolution, and you can actually use this instead of assuming a hemispherical apex during the reconstruction. And theoretically, this can correct for distortions due to evaporation field differences. So this is still quite early work, but basically by calibrating the model times to experiment times, so the index, the sort of ion arrival time, or the index in the sort of evaporation sequence, and then basically making some assumption about the model geometry or your specimen geometry, you can correct the reconstruction. So there's this sort of there's this narrowing in the central channel within the standard point projection reconstruction, which is due to a low field phase down the center of the central channel. And then basically using the model, you can correct for this once you've calibrated the fields and the times between the model and experiment. So setting up the tool is fairly simple. So the code can be found here. And then basically this can run on both Ubuntu and Windows 10 using the Ubuntu terminal, which is available through the Windows App Store. The tool can be directly used from the command line or as a Python package. So the back end is written in C++ and compiled Python. The front end is Python. And then, yeah, it's fairly easy to install and set up. So README is provided. The installation pretty much consists of 2 lines. One just to install the dependencies of the package and the other one just to compile the code. And then introductory tutorials are also available as IPython notebooks on the repository. So these cover simulation setup, running of simulations, and tools for data processing and reconstruction. So in summary, Casra is a tool for simulating and solving distortions in atom probe experiments. It allows for fast simulation of realistic specimen shapes. And basically these models, another benefit of sort of these simulations being sufficiently quick is that you can use these models to directly drive the reconstruction process. Thank you for listening.
Okay, so are there questions to Schatz on the line? Yeah, great talk. Thank you. Thank you, Schatz. Are there questions to Casra, the use of Casra and the integration into tools? Yeah, there's one from Benjamin Kaplans.
Is there a question? Sorry, what's the? Can you model voids? Yes, you can. So basically, the method of simulation uses something called the level set method, and this allows you to basically track or model the joining and separating of curves or surfaces. So yes, this basically allows you to model voids. And actually, I've got a paper that's in press. I think it should be coming out sometime in the next 24 hours, at least the accepted manuscript, which talks a bit about modeling voids using Casra. But yeah, that is definitely something you can do.
Can I ask a question? Are you planning on doing any kind of modeling of the detection process?
So not at the minute. So at the minute, I've just been focusing on kind of the distortions that are arising. But yeah, I guess you could model the detection process. The other thing, I guess, is also investigating kind of a generalization for the Ball correction. As in now we know what the shape of the emitter is. You might be able to try and correct time of flights using that information.
Yeah, that's actually a really good idea.
So can you do iterative reconstruction to minimize error? Comes from Andrew Protein.
So not at the minute. So currently the model basically assumes the geometry. So you can iteratively reconstruct varying the evaporation fields. But at the minute the geometry is fixed, but you could in theory do a similar procedure where you kind of what's called shape optimization, where you basically change the geometries of the phases over time and then try and match up the reconstruction to the model you started with. So yeah, you could do it, but I've been looking at iterative reconstruction, varying evaporation fields, but just keeping the sort of the specimen geometry fixed at the moment.
Can I have a question?
Yeah, Peter. Yeah.
A lot of the level sort method would depend on the quantization of the surface, essentially directly on a triangulation. So how do you make sure that a triangulation is fit for an accurate representation, essentially, of the field distribution on the surface? Because I can see you have some adaptive meshing here, right? At the shank, it's a little bit coarser.
Yeah. So I'm using Seagull to do the adaptive meshing. And it's basically, it runs as an isotropic remeshing algorithm in Seagull. And that basically ensures that the mesh is well conditioned for the electrostatic solver.
So it's based on an octree then, probably.
Yeah.
Is that what it does?
Yes, I think I'm not exactly sure. But yeah, I think it's based on some tree-based method. And then yeah, the isotropic remeshing, you can kind of remesh at different sizes down the shank.
Have you had a look in the numeric variations at sort of the larger elements? What kind of variations would happen there? Because there could obviously be in pinch points, you could have numeric instabilities, essentially.
Yes, well, I have a little bit, not in too much detail, but yeah, it is something that does arise, that if you have too big, if you sort of your triangle size increases too rapidly down the shank, then you do end up with a very poor quality solution at the apex. So yeah, it becomes very poorly conditioned, the problem.
Okay, so
but yeah, the tricks.
Yeah, just make.
Go ahead.
Go ahead, Charlie.
Oh yeah, I was just saying. So at the minute, I'm kind of just manually tuning it to ensure that the sort of the solution near the apex is well conditioned.
Great.
So
I think we should move on with that, Marcus.
Exactly.
If that's all right.
Exactly.
Great. Thank you. Hopefully.
Dear Atom Probe community, my name is Markus Kübach. I'm a postdoc at Max Planck Institut für Eisenforschung and also a member of the Atom Probe TC committee. And today it's really my pleasure to present you my ideas about how we as a community could become even stronger in aligning our data with the fair data stewardship principles and which consequences and ideas this might provoke for software development. I'm going to start with my perception that many of us in the community feel that Atom Probe software could serve us much better. So we need to invest our time wisely, and that is exactly why we need to use productive tools. And software development and FAIR is one such tool. The key statement that I want to make today is that Atom Probe software development should be a community effort. Some of you would argue we've done so in the past. I agree, but we could have improved much stronger in terms of avoid re-implementing the wheel. Focus on specific tools, on our specific expertise, and not try to cover everything. So what are the fair data stewardship principles? And why the heck should I care? The fair data stewardship principle in fact state 4 very somewhat abstract ideas that need to be made concrete by implementation and living by a certain community. In our case, I think our community, the Atom Probers. Data should be findable, data should be accessible, Data should be interoperable, and data should be reproducible, i.e. repurposable for a different task, completely different to what you might have thought your data set might be useful for. So people should be able to find where a data set is, should be able to understand what the story behind that data set is, with as many details as possible. And if you ever want to train a machine to do some machine learning on your data, then you better train the machine and you better inject the metadata such that the machine has the same wisdom as you have on your data set. But by the end of the day, if a machine is ever to analyze a certain data set, it needs to understand the data format. And ideally, we as a community want to support and want to be able to parse content from such data. And therefore, I think the only alternative are open data formats. So I want to quickly summarize the status quo on workflows and metadata in AtomProbe and FIM, so related techniques. We know all that AtomProbe is essentially a combination of workflows: specimen preparation, measurement, data post-processing or analysis of the data, publication, and then curation of the publication. And I think it's important that we not only focus on the measurement side, only on the data post-processing side, but by the end of the day, that we take into account that all this is coupled into a large workflow with individual pieces, and ideally all of these should be fair. However, if you look in reality at the moment, there is a plethora of tools that we use and a plethora of approaches how we store information with respect to something which by the end of the day ends up in the same figure in the same paper. With the last slide, I want to answer a little bit on the idea, how can I personally contribute to the ideas I've just mentioned? So development of complementary tools and approaches. And for this purpose, I developed one contribution. And one contribution so far is ParaProbe. This is a set of open source software tools, with scientific computing technology in the background. The idea of the software is quite simple. You define your workflow, and that is essentially, I have a data set, several atom types I want to process, several advanced data analysis techniques I want to execute. On all these data sets, on all these possible combinations, then ideally what we do in computational materials science, we do batch processing, of course. Yeah, we would never do this manually. And that unfortunately is not so easy with AtomProbe, and that's exactly what ParaProbe solves. Yeah? So you define in Python what you want, then Python essentially creates the necessary input files. You take these input files and your data sets, go on a laptop, a workstation, a supercomputer, whatever resources you have available, and then the processing is done in the background using performant hardware, whatever you have there. The benefit is everything is open source, so you can dig into. The second benefit is everything is documented, also the metadata. So I've invested quite some time to document what the metadata are. There's definitely not the last shot on it, and I would be very happy to discuss this with you as a community in particular. And yes, this is the idea in a nutshell. Yeah. So open workflows which use all CPU cores, whether you use a laptop, whether you use available resources, whether you have a supercomputer. And if you have 1,000 CPUs, you want to use the 1,000 CPUs. If you have 100 GPUs, you want to use the 100 GPUs for your task. Yeah, many people can do that in many scientific communities, and that should be also possible in AtomProbe tomography. And that's why I invested time into this. With that, thank you very much for your interest, and I'm very happy to discuss with you on all sorts of aspects that you've raised also in your talks. Thank you very much.
Yeah, thank you very much. If you look the entire video without recording something on Zoom, you might also see that it's perfectly in time and not lagging. Okay, are there questions?
I had a quick question. Before you run your job basically on the supercomputer, how do you know what you want?
You essentially define what you do beforehand. So the tool is rather meant to process combinations of batch processing, combinations of multiple data sets. So essentially for explorative analysis, and you define this beforehand using a set of Python classes. So essentially you write a Python script for which there are tutorials online, and then you create the necessary input files. So this is all hidden in front of the user, such that people have not to wrangle about with text files and so.
and then you take a text file, run on the supercomputer or on your laptop or workstation. And at the moment, we are working on integrating this into Anaconda, such that people can just download and run it locally. But that's essentially the idea.
That's great. Sounds excellent. Thanks, Marcus.
I have a question for Marcus.
Sure.
What are your in and your out points for data? Because so far I've only seen sort of the things that have been floating around in AtomProbe, EPOS, and whatnot. But what would be your in and out points?
So my in and out point is essentially, I say one task, one tool. So if you, for instance, make a reconstruction, you go in with your file, whatever it is, APT, EPOS, or POS. That's the input. You do the processing and you get an output. That's a file. And then typically what the next, if you want then do analysis on that, you have the next tool parsing from that process file. So essentially you haggle through your analysis, guided through a set of files which are all open and metadata documenting. There's a little bit the trade-off to make between making it performant, on the other hand, handle large data sets. Of course, there are certain analysis combinations for which it would be smarter to keep everything in main memory, substantially faster. But essentially in the sense that you typically handle only 2 billion points at most, typically in AtomProbe. This for cluster or so workstations is not so much an issue IO-wise. So typically that's why I say you start, you hand data over via files, and if you look into the if I look into the data, how much time typically the IO takes of this compromise, I have to say, then it's in the order of 1 or 2 persons. So that's typically negligible. Yeah. But yes, one task, one tool. So you hand in the result and you pass it to the next stage. And that gives you the flexibility.
You can pull it out at any point.
Exactly. And it's also documenting each step. So also the metadata in the HDF file are documented, so you know exactly what you have done. And that, of course, gives you the flexibility to then parse together the pieces that you want, and also essentially gives the flexibility, if you have other tools, whether it's Python, whether it's MATLAB, to inject code from something else.
So you're using HDF5 to pass it on?
I'm using HDF5, exactly. Yeah.
Okay, great. I think with that, we will... I'll ask Peter, can you share your screen?
I will share my screen, and I want to thank Marcus for doing a perfect setup of what I wanted to show today.
Great.
Because what he's been showing has actually been near and dear to our heart for quite a long time. And so in the last period of time during COVID, we've really consolidated all of the code that we had around AtomProbe analysis. And so now we are able to, can you all see my screen?
Yeah, it's looking good.
Yeah, okay.
Great.
So we've put together an entire AtomProbe analysis package that gives you all the tools essentially that you need to analyze your data from beginning to end in an interoperable way. So I agree very much that we should be able to at any point jump from tool A to tool B without any problems. And we sort of give you the basic analysis tools that you need to cover everything that that is sort of the basic analysis things. And we cover it in a way that allows you to be very interoperable, and that allow you to create what I call institutional knowledge. Yeah? So you're starting to build up certain what we call ground truths, and based on those ground truths, you work your way up. Our most basic ground truths are actually an isotope table. So you can see we've got a table of isotopic abundances and weights and so on and so forth, and a color scheme. Yeah? So you give a color scheme out for certain ions, and you tell it what kind of color you want it to be. Then you can go to ranging. So you can do all of the ranging by first defining ions and then defining ranges on top of them. And we give you the full ability to search for ions in your code. So you can see here as a quick example, I've got a superalloy mass spectrum, and I can go ion find, and it will pop up with the mass spectrum. I can mark a peak, and it will tell me, okay, this is most likely chromium-52, but I could try out if it's something else. But I guess chromium-52 was a pretty good guess by our programs, and I can go okay, and then just say, okay, add a range in mass spectrum number 2. Sorry, because I've got range add, sorry, range add. And then you can just go and mark your mass spectrum window, and it will show you, okay, this is chromium-52 2+, and you can assign all of that in your to your pos variable, and then your pos variable will show you for each hit what the XYZ is, what mass to charge, what ion it is, what charge state, what atom, what isotope, and what ion complexity. And if you have more information, can give you a whole lot more, but this is just for a regular POS file. And then you can put your analysis on top of all of that. And we offer you quite a lot of different analysis tools, most importantly, advanced concentration analysis. So you can bin your data in n dimensions based on whatever property you like. And then you can run what we call concentration kernels on your data and calculate things like, in this case, a 2D concentration map. And you can use all of the information that you have in your mass spectrum about ions, about ranges, and so on and so forth, to do that analysis. And it's all at any point accessible to you. And it's not just accessible to you if you want to use MATLAB, but you can also go outside of MATLAB and export it. And just like the group at NPIE, or at least like Marcus, we're using HDF5, and we've put quite some effort into creating HDF5 definitions in order for your data to be very shareable. I have to say, in this case, our starting point was not atom probe tomography, but we have big collaborative research centers that require us to have data and metadata together. And atom probe tomography is just one very small aspect of that. There's SEM, there is TEM, there is EBSD, there is particle analysis, there's electrophoresis, and so on and so forth. And so we've come up with metadata definition files like this file here, where we have path and where we define the naming of whatever metadata we have and of whatever data we have that we can then inject into our HDF5 file. We can do that for atom probe data quite easily by now. And you can see you get a file like this, which you can, for example, open with an HDF5 file viewer. And it will show you what information is in the file and what you can do with it. So you only need the file. You need no explanation from us, okay? If someone gives you the HDF5 file, you need no information. What's in there, you can find all of that out for yourself. In this case, we've defined a reconstruction. We've defined images of the sample. So I can, for example, open the image of the sample here. Sorry, I'll just go and show the data. And you can see it's an SEM image, right? That is saved together with the atom probe data. This really allows you to build up highly capable data science analysis for atom probe tomography, because we've made everything as scriptable as possible for you to be able to do your analysis not on one data set, but on 1,000 data sets or 10,000 data sets. Okay, and yeah, that's it from me for today, because I've already talked for 6 minutes.
Peter, great. Really supporting this. Should discuss more about this at some point. Are there questions to Peter's talk?
Oh, by the way, the code is all available to anyone that wants to use it under I'll give the GitHub link. We will publish something about it, but if you're interested already, you can go on it. It comes with all kinds of tutorials and MATLAB live scripts that just explain everything and how to use it. My students can by now use it by themselves, so yeah.
Great. There are no questions at the moment in the chat. There's one popping up from Philip. Does your code have any overlap solving built in at the moment?
Not the code that we've added to the GitHub, but the purpose of explicitly defining isotopes and everything in the mass spectrum, so defining all the ions in the mass spectrum, is actually explicitly made to deal with overlaps, yes. But the what we call concentration kernel for overlap solving is not yet pushed into the GitHub Atom Probe toolbox, but it will be in the coming months. Yeah. And the good thing is you can use that concentration kernel for any dimensionality. You can use it for the entire data set, for a proxygram, for a 2D map, for voxelization, for whatever you like.
Okay, this seems to answer the question. Further thoughts and ideas and questions to Peter? No reason to be shy.
And also we also have very extensive code to do machine calibration, because we do have 2 atom probes that we've built ourselves, apart from the commercial one that we operate. But this will only be included in the next release, probably early next year, because we sort of, it's not as user-friendly as the other scripts in there.
Do you have any plans, Peter, to do anything for helping people get started with this?
So right now there are already tutorials in the form of MATLAB live scripts in there. Let me just show it to you. I can show you one what that looks like. So this is a where did I put it? Where did it go? Oh yeah, this is a MATLAB live script. Yeah, and this live script, for example, explains how to create an isosurface. Yeah, with either just programmatically or using the isosurface widget app that we supply. But my student, Martina Heller, is currently recording video tutorials that will be available hopefully not too long in the future. But it should be possible for people to get started based on the MATLAB live scripts already.
That's great. Thank you very much.
The point is, this code is not meant to be a feel-happy graphical user interface. It's meant for people to be able to do analysis on a lot of data in a short amount of time. Again, we want to see not analysis on 1 data set or 2 data sets. We want to see analysis on 1,000 data sets, so that we can see trends. What does the background in Atom Probe look like depending on the pressure in the measurement chamber? Yeah, things like that. But also all your standard analysis is included as well. This is 10 years of me coding that's included in there.
Yeah, no, I know the feeling very much, and I'd say my own work, the transition from looking at doing analysis on a single file at a time to being able to run a bunch of stuff all at once is definitely very empowering. So thank you very much for that, Peter. Right, so next up we have a short 10-minute presentation about the current outputs from the Atom Probe Technical Committee, and hopefully this will lead on to some discussion as well as mopping up some of the questions that we've had to skip over until now. So Dan Haley, if you could take over, you should have control of the screen.
Great, thanks, Andy. So it's really good to see that there's a lot of grassroots efforts throughout the community to try and solve a lot of the analysis problems that we're seeing. And I think that gives us a really good introduction into why we need the technical committee and the activities that we're undertaking. So let's just try and walk through some of these outputs that we've generated as a technical committee. And I would say one thing to me that I think is really important to emphasize is that there's a lot of disparate efforts, and trying to bring them back together is one really important task. And that really helps everybody speak a common language. And 2, it's actually a surprisingly tricky task because there's a lot of different needs throughout the community, and everyone's has different sort of backgrounds and where they're coming from. So having the TC as a vehicle which we can use to try and focus our efforts, I think it is a key goal. So let's just talk about some of these efforts that we've gone through. So hopefully I haven't scrolled too far there. Yep, I have. So this first thing we had a question from the audience actually was, do we have a sort of way of being able to keep track of what software is available? And so that was one of the first things the technical committee identified as a need for the community, was there's a lot of these disparate tools out there, and various people can rattle off which ones they are, but very few people have a complete listing of what's available, what their status is, and a sort of, I guess, the metadata around each of those pieces of software. And so what we've done is we've brought that into a single spreadsheet, an online Google Sheet that the TC maintains and will continue to maintain, that lists all of the software that we were aware of up to date. Obviously, there's a few new ones that are coming out now, and we'll be adding those. But this is a resource that if you want to know whether or not there's a tool within the Atom Probe community that undertakes the task that you need, you can have a browse through, visit the hyperlinked websites, look at the sort of code languages, whether they match your needs, are they being actively developed, these sorts of things. There's some data about various functionalities. That's not fully complete in terms of, we can't map out every functionality of every tool, but it gives a snapshot of what's available across the different software packs. So you'll see that software is actively maintained, and we are going to keep that going forward. If you are developing software and you have released it, I think we need to definitely hear from you guys in order to be able to push your software onto this list. So a lot of the discussion we've had today is about data identification, sort of doing things like discovery and provenance. So what data do I have? How do I find it? And where did this data set I currently have in my hands come from, and what was in it? So we need to be able to enable reuse and discussion. So we need to have the way of being able to describe what data is what. So this file I have, what is it? What is this .blah file? I've never heard of it before. How do I open it? What do I do with it? Who made this file? Okay, I have a question about how this work was conducted. Maybe there's some metadata missing that I need. How did you wash your samples or something like that? Can we go back and be able to reconstruct these questions readily? How do I know the data is actually in fact correct? How do I know that through various transformations or archiving or whatever, it hasn't been damaged or disrupted in the process? And say I didn't have a data set, but I'm looking for one, where can I find where these data sets are, or where did it come from? So we need to have a way to be able to determine where data come from, and to be able to have a way to be able to read and write and describe data. And I would say at the moment, unfortunately, we don't really have a way to be able to exchange data commonly between various labs. I think there's a lot of de facto ways of doing this, and they're not necessarily providing complete coverage across the community. And so what we're trying to do is provide some work for that to back up what people are doing. So to be able to do this, one of the first things we identified as a relatively straightforward means of doing this is a technique called hashing. And this answers a few questions which I'll get to. But firstly, describe what a hash actually is. It's essentially a file fingerprint. If you take in the file in terms of its contents, ignore the file name, and you run it through an algorithm, you can basically generate a fingerprint for that. And it looks like a bunch of letters and numbers. Okay. So what we could say is that, each of these fingerprints individually describes a very specific file. So that file is going to have to exist. We're going to have to know the contents of that file, and we can match that back to the original file. So we could eventually, instead of having to reproduce the entire methods within that, which might be incomplete within your method section of your paper, because they're quite short, we could just provide the information as essentially a part of the fingerprint, and you could retrieve that either potentially automatically. It doesn't really depend on how you choose to name it. So one of the advantages of hashes is that they're distributed. You will need to have the file in order to be able to create the hash. And so the other advantage is that the hash as a fingerprint is one way. So you can't reconstruct the contents of the file from that set of digits. There's a huge amount of information within an Atom Probe run, and we've managed to compress it down to something that fits across my screen. So there's no way to reverse that. So that's important from a commercial sensitivity point of view. A lot of labs are publishing or analyzing data which is potentially commercially sensitive. The hash protects that. And so you can still publish your hash of your data even if you don't want to publish the data itself. And so you can also guarantee that a particular file you might have in your hand matches that. So let's just walk through a quick example here. So some of the benefits we've got is the fingerprint proves the existence of a file. So if someone has pre-published the hash, you know when I generated that original data. The fingerprint is also vendor neutral. It means that because I'm only looking at the contents of the data, it doesn't matter how you structure that data. It doesn't matter who created that data. Anyone can generate a hash at any point in time. So it's vendor neutral. It works anywhere, it works anytime. It only takes a fraction of a second on any computer. You can really build those up very quickly. And so you can prove that a data set exists even before you publish the contents of the data set. You can say that I have data set one with hash 1234, And later you can come back and say, look, here is the data set 1, 2, 3, 4, and here are the contents, the one that I said in the paper, and I was under embargo for 12 months. And you can prove that those actually directly match to each other. So it provides a unique and very clear tracking mechanism. And this really helps with compliance with FAIR. It's part of the findable part of it, because I can research directly using that hash. It's part of the sort of reusable bit, because I can track back to where that data came from. So as I said, the hash is only one way. So let's try and recover some data. So I get a blob that someone sent me in my, link to an email, I download some file, and someone's called it experiment2.blob. Very helpful. What is experiment2.blob? How do I open it? Where did it come from? So let's look at the hash. Okay, so here's, I run the hash algorithm and I generate this fingerprint. I then search through a set of text files that are either available online or on my computer, and it shows up in the Oxford Atom Probe group listing. So if you were to search for this in the appropriate location online, you would in fact see that this turns up in Oxford's published list. And it was experiment R83-06086. So I didn't have to know anything about the file. I didn't have to worry about the contents or the name. I was able to directly match that back to both Oxford's experiment list. I know that they did that on the 9th of June or before, because they published that hash on the 9th of June. I know which experiment it is, and I know that the file, importantly, is intact. It hasn't been damaged in any way, because I can match that back to Oxford's list. So what we're saying as a TC is we would like to try and promote groups publishing their hash lists of their data set. And we've made software available, thanks to our students at Oxford, who are providing software to be able to do this. So any lab can simply just run that Python code, and it will generate a complete hash list of all of your data. You publish the hash. You haven't published your data, but you can prove exactly where that data set comes from. And if someone grabs a random data set, they don't need to know that R72 matches back to, I don't know, let's say Sydney. I don't think it's Sydney. And then I can contact the city people and say, by the way, I have R72 experiments on your list. Can you tell me a little bit more about it? So the next task we identified is this open data format. So a lot of labs at the moment are coming from different vendors. They're either building equipment themselves, they're working with other vendors that are disparate from various locations. And we need a common way to be able to communicate with each other. And so we've been working towards developing a standard which uses the HDF5 file format, which is a well-established container format, that will provide a vendor-neutral way to be able to communicate between labs. And what it's recording is essentially the machine output from an atom probe experiment. So at the moment, we're terminating our work at what comes out of the instrument. And so that should contain all of the information you need in order to be able to reconstruct and analyze the data set that came off that instrument. It will link back to sample databases. It will link to run conditions. It will link to which labs are running those experiments. All of this information will be available. And so that's a draft at the moment. We're looking to try and complete that and make that available soon. We've had an initial round of going through this, and there's a few more suggestions have cropped up, but we're hoping to complete that shortly. And so we've had agreement from various vendors around the world that they will help support this format going forwards. And so you'll either be able to use directly or import this into the software packages that you make available, that are made available. So in conclusion, what we're trying to do is to make all researchers' lives easier and to try and remove tedium. We're trying to create standard identifiers for data. We're trying to make things more discoverable, and we're trying to create reusable containers and systems to allow researchers to work with that. And this will maximize the benefit of what we do in atom probe research. And minimize the amount of time that we have to spend trying to understand our file formats or code or write things and be able to work together in a common language. And this really is at the heart of FAIR, to be able to make research reproducible. The TC, in terms of issues that we see going forwards, is very happy to be contacted from the community. So you can either do that through your local representative or through the TC. And we're here to try and work with you to be able to solve issues that are important to you. Okay, thank you very much.
Thank you, Daniel, for this great wrap-up of what the TC is doing. So now we're moving into the complete discussion slot. So you can now fire up all your questions you have.
Yeah.
I would, just go about and let's see if we get some structure in and wrap up on the questions that we had in the chat and haven't answered.
Yeah, so I think we have a few questions outstanding at the moment. So users who are still working on getting their questions together, just do that now. But I think we had a question from Andrew Prudian that relates back to some of Tim's stuff from Kamika.
Yeah.
Does the APT file format contain all of the data that resides within a HITS file? Tim, if you're still there, there's a question for you.
Yeah, I am still here. I actually responded privately to that one, but I'll respond for everybody here. Optionally, you can add almost anything from the HITS file to the APT file. It's optional, so you don't have to waste disk space for data that you don't actually care about in your reconstructed data. And there are some internal debugging type things that you can't put in the APT file, but in general, all the things that anybody doing research on Atom Probe would need are available.
Can I ask a question off the back of that, Tim?
Sure.
Is the timing information available there? So not just the XY time of flight, but the unresolved timing information?
You know that's actually not even in the HITS file, so
okay.
So no, it is not available.
Okay.
Yep.
Thanks. Dan, were there other questions you'd been able?
There were.
I'm
just having a quick chat through them here. So we've had one. I think it was from Martin Meyer at Oxford regarding Jens. Jens, are you still on the line?
Yes, I'm still here.
Okay. With your code, is there a specific need for certain MATLAB versions to be able to support that? What versions of MATLAB going forwards are you supporting?
I'm not 100% sure, but it should be something like 2016 upwards. Then there should be no big problem.
Okay. All right, that's good to hear. And we have another question, and I'll run that. Well, actually, Andy, do you want to run through this question from Ben Kaplan about the Atom?
So does it currently have time of flight analysis and algorithm things, voltage and ball corrections? Does it function more on POS type information? That's the right question, Dan.
Yeah.
Yep. Yes, at the moment, LibAtomProbe is primarily focused, I think, on the POS type information, but it does have the capability of reading in that kind of raw data if you have it available in a format that LibAtomProbe can open. So, for example, we're not able to open proprietary software formats from Kameka, for example. I think voltage ball correction, we don't have anything, if that's right, Dan. I know I've done my own thing in MATLAB. It can be relatively straightforward. It depends precisely how you want to do it. So a simple correction is fairly straightforward. If you want to do something that's maybe potentially more accurate, then your algorithm has to get more complicated. We don't have an algorithm that does that. We'd be happy to include something. I think LibAtomProbe gives you most of the basic tools you need if you just had X, Y time of flight to then turn that into an actual reconstruction. But we're not working on data. We're mostly working on data that's already been reconstructed. So I think there's a good opportunity there for working with Peter and Patrick, etc. To try and get that full reconstruction chain actually there as a basic tool that people can use. I think there's just not been the need for it at the moment.
We certainly have that available. The thing is, right now it's not in, like, we've really polished all of the scripts that we've put on the Atom Probe toolbox. That's not really polished, but if you want to have it before, and I can give you voltage and ball correction tools. It's not very complicated to do voltage and ball corrections. It's not hard. It just needed to do it iteratively because the drift regions aren't field free.
Yeah.
We do have one Atom Probe that is completely field free inside. Then it's completely trivial.
Okay.
I'll just piggyback a comment of Andy. I think one of the goals that we're trying to do with LibAtomProbe is to make it sort of there are a lot of different modules. And so as Andy says, a lot of our focus has been on trying to do the modules that are important to us. So it's very difficult to know what's needed for the community. We need to really get a bit of feedback there about what people are using, what people need. There are other functionality within that. There is other functionality within that software, things like mesh processing, spectral analysis, working out background counts, doing things like ion identification, where it's not focused on POS files. So I think it's quite a horizontal library in terms of it covers lots of different things, but it's very needs driven. So you might find that it doesn't necessarily capture a need. But we'd definitely like to hear about what your need is and what we could incorporate there.
So Aaron tries to point a little bit to these rather broader type of discussions and comparison of tools, where he asks, given there's a lot of different programming languages, software packages, etc., used by different researchers here as presented today, is there any discussion to figure out which tool is best for a specific purpose or technical question, or essentially comparison of performance between these different tools for doing same analysis, for example? And I think this is an excellent question, pointing to exactly to something which hopefully FAIR will be one tool for to solve. What would be the point that the individual programmers and tool developers would like to raise on that, to Aaron's question?
Well, I'd just like to say that there's work, kind of a subgroup, including other people as well from the technical committee led by Anna Tugara from Sydney, on exactly this last point, comparison of performance. And here we're not talking speed performance, but in terms of accuracy of different tools. And so Anna is very much at the moment leading an investigation into that, specifically comparing different data sets, real data sets, simulated data sets where the ground truth is known, seeing how different algorithms come up with different answers, maybe exploring edge cases that might come up, dealing with, computational issues as well as more generally applicable atom probe issues, so I'm helping out on the composition quantification, for example, and whether it's necessary to use sort of full peak fitting in order to pull out composition, or do we just need to use ranging? What's the influence of local background subtraction? Does it matter how you do that? These are really important questions, and they really influence our analysis potentially quite strongly. And at the moment, we're kind of flying blind in terms of knowing where the problems are. And so I think that's what Anna's trying to address with that. I'd love if other developers wanted to chip in on what they thought was their best programming language for certain approaches. Just with my Atom Probe, LibAtomProbe hat on, I'd say I would hope LibAtomProbe would be portable enough that anyone could use it in any kind of analysis. I know that there's compilation, integration issues that can make it a bit of a headache, say using it with something like MATLAB. But we would love it to be, as a building block that everyone could build on, so we can avoid this problem of everyone and their dog having to write their own range reading file format, for example. But yeah, does anyone else have any comments on what they think is the best tool for the job for different analyses?
I'm happy to chuck in. So that's my very personal opinion. We should stop asking questions like, what is the particular best programming language for doing a certain job? So you use a programming language that you feel comfortable with. What I personally am also participating in this research with Anna find most impeding on getting forward here is that how can we interface with the work from others, document what other people have done? And I think we should rather ask how we can make this more explicit, and ask how can we, if we have to program this, how can we make our job easy there in the programming without sacrificing rigorousness? That would be rather the question I personally would put forward, instead of which particular programming language. Peter.
Yeah. I could not agree stronger with Marcus. So the question is not really which programming language. So the history of the Atom Probe toolbox for us was I started it in Python, and I moved it to MATLAB because widgetification, so writing little widgets in Python, is suboptimal. That's why from the beginning on, it was planned to, at every point along the way, to put everything into an externally readable container. And I think we all came to the same conclusion. Marcus, Dan, everyone, we all came to the same conclusion: chuck everything into HDF5 containers, so that anyone at the point when they have the tool that they need can pull out the information. And I think that says a lot, that independently we all came to the same conclusion.
Yeah, I would say there are definitely decisions like that that get made that are very practical, like eminently practical. Like, I want to make a pop-up box and I can't do it in X, and I can do it really easily in Y, and so that's why we make these decisions. Yeah, that's definitely a key driving force.
The way that we've approached it is that essentially you have a base HDF5 file, and at every point at the analysis you can add to it, right? So for example, in the beginning, we start out with your experimental metadata and your hit data. And then you might throw in an SEM image of your sample or 10 SEM images of your sample. And then you chuck in what ions you have, you chuck in what ranges you have, then you chuck in any ROIs you've defined, and so on and so forth. And that all sits in there. And it's especially important for us because we have done the same for EBSD, for creep data, for particle preparation things. And so you can now go, or you will soon be able to go into our server and say, give me all data from aluminium where I have on the same sample where I have creep data and atom probe data, or superalloy where I have creep data and I have atom probe data and EBSD data, so that you actually can use your data in 20, 30, 40, 50 years, and you have 1,000 data sets. And this is really the purpose, and this is really the marching direction in this case.
Yeah, that's great. There's a, sorry.
Geodata. There's one comment from Geodata.
Yes.
Yeah.
Do you wanna...
Okay, great. Yeah, great.
Yeah.
No, just go ahead.
So there's a question about potentially a round robin of sharing data and allowing different users to treat the same data with different packages. I think speaking as the TC, I don't know whether that's something we would really want to be involved in. We would love it if the community organized that kind of engagement. I think it would be in some ways similar to what Anna was doing, but I was involved with a round robin with Emmanuel Marquis looking at clustering, and it was a really eye-opening experience. When you ask a dozen people to do the same atom probe analysis and you get basically a dozen different answers, and then you try and unpick through this really convoluted analysis chain, why were these decisions made? How even do we just simply share the kind of output data? It was tricky to even agree on those kind of simple things, let alone get to the point where we could say, all right, we actually trust the maximum separation algorithm for doing this kind of quantitative clustering analysis. So yeah, I think that kind of round robin initiative is really, really great. And I would love to see more of that happen, especially, with certain tools. I'm thinking, composition quantification. As we've seen, there's quite a few different ways of doing that. I'd love if we had, the same sample. We could all run it. We could even have simulated data and analyze it. I think that would be great. But we're already trying to do quite a lot of the on the TC. We're fairly thinly stretched. So we would love to enable the community to do these things as much as possible. That would be my. Hopefully, the other TC members are on board with that. We want community buy-in as much as possible.
Yeah, I just want to back up what Andy's saying there. I think that the value of these efforts is incredibly immense. But I would say a lot of the people who run these round robin exercises find it quite tricky to be able to pull all that information together, and they take quite a long time to set up, sometimes several years, to be able to do things like running the same sample in a couple of different machines, or getting everyone to back. I think the issue is we're busy researchers, and the sort of round robin question is quite easy just to push down because I need to get this analysis done before the end of the day. And so over time that builds up into quite a long effort. And so I think the technical committee, before we go down the road of trying to explore round robins, this might be something we would consider in the future, is to make sure that we have interchange, because I think that's part of the problem with the round robins at the moment, is data interchange is still a challenge. So that when you have large groups of people, you actually find out we're speaking slightly different languages, and we get a little bit of confusion arising from those. So to be able to make those round robins easier, I think we need the ground levels of communication between research labs really well exercised, so that moving data backwards and forwards is not something that requires 20, 30 emails to be like, oh, actually, I'm missing this piece or that piece or whatever. It's just, here's the data, here's the server, I can just grab this. And even in some cases, we might be able to do that retrospectively. So if the data is made publicly available, it might not be that you actually have to do an explicit round robin. There are a lot of aluminium materials that are analyzed in the world, pure aluminium, pure silicon, or near pure silicon, because these are relatively standard samples. So you could imagine that if we were all to publish our data, we could actually run these round robins retrospectively, but we need a way to be able to communicate that to one another before we can do that. And so being able to enable this is something that FAIR is really building on, and something that is tricky at the moment, but with a bit more groundwork could become easy in the future.
That's great. Arun, I think you had a question. Do you want to go ahead?
Yeah, so not a question, but just a suggestion. So this is fantastic. This was probably the first time I've seen a nice collection of all the different tools and things being developed in one place. So one thing which you can think of is there will be an M&M, so Microscopy and Microanalysis Journal special issue for APTNM. The deadline for it will be sometime in August. So basically 1 month, actually 1 month after the APTNM rescheduled date. Your Excel file showing that compilation of all the different tools and developments going on is a fantastic start. But have you guys thought of writing a comprehensive review article where, the contributors for today's workshop seems like a fantastic collection where if all of you writes some detailed description of this and a very good introduction like what Dan and Andy talked about, that may be a very good review article to consider for the APTNM special issue. So that was kind of the suggestion I had. And the second thing is, even in PNNL, between me, Dan, and Danny, we have been looking at the total number of data that we have been collecting over last 10 years since the Atom Probe came here. And more recently, Jing Wang, who is actually here online, is trying to essentially do the same thing where collect this probably several thousand data sets and look through specific collection of data where we have analyzed laser energy versus stoichiometry, for example, and then trying to analyze these. So a lot of the things that were discussed today were also useful. That was basically my comment.
Yeah, it's kind of related to that. There was sorry, my son's in the background. I don't know if you can hear. There was a comment regarding whether we could have some kind of metadata database that people could use. We did look briefly into this. The main thing is we would need community buy-in. It would only work if people were happy to submit to that. But if people wanted to send in data to that database, then that would be great. And we were initially looking at what was currently available and whether or not we could set up our own, whether we were able to host our own database or piggyback on some other service. But yeah, I think that would be kind of take our analysis along to the next step.
Mm-hmm. Okay.
So see, there's another question from Torben Bull about hash identification and whether or not people are going to buy into this. I think this piggybacks off what Andy is saying. A lot of what we're doing here is to test the waters of how much buy-in we can really get from labs. And on my personal side, I think that ultimately we don't need to force anybody to do this. Your funders are going to do it for you. So within the UK, if you do not make your data fair, it does not count. If you have a publication and it is not fair, it essentially counts as negative points for future funding for your departments. So department down or university down, they're pushing that we have to do this. So it's part of the Act on Acceptance requirements from our university is that once we get a paper published, if we want this to count towards our future funding, we have to start saying, okay, we actually did this and we made it open access and we made it available. Currently, it's only focused on papers, but for the next REF 2021, they're talking about data. So if you're not making your data available in such a way that you do not need to contact the author ahead of time, then you have not published your data and your department will have funding removed if you don't do that. So the EU, the US in some areas and the UK are working towards making this a requirement for researchers. So I think for senior researchers, I would agree, this is probably less of an issue, but for junior researchers, you're going to start seeing this transformation slowly creeping forward. And I think that the line that the UK is using is public funds, public research. If you are a publicly funded organization, you're going to have to make your efforts public. And so what we're providing is a tool for you to be able to do compliance very trivially. So you just run this program against your database, and all of a sudden, boom, you've published your data, and it's there, and it's findable. You don't have to do any effort. It just works for you.
Yeah, so I mean, there's one of the main areas the TC is trying to tackle. Is just trying to make that process as easy as possible. In one of the key things for that is identifying and recommending a standard. So having a standard is really important, because if someone looks back on our AtomProbe data in 10 years time and it's in some random file format, that's as good as useless. So it's not just file formats, but standardization of metadata, and it goes the whole way. But we want to try and put in place systems to make this as easy as possible. So we're not going to be able to do all your work for you, but we want to be able to work together and bring the unity in the community as much as possible. And that's challenging, but yeah, we'd very much, we're here having the discussion, so that's a start.
Yeah, and maybe a comment from me on that. So as long as it's a small finite number of different file definitions, I don't think it's a big problem. If I get an HDF5 file from Dan and it's slightly differently structured than mine, or from Marcus and it's slightly differently structured than mine, it takes me about 10 minutes to write a converter. What I need to be able to do is to see where things are, to open it up and see where things are and how they are structured. As long as I can do that, I don't see a problem. As long as we don't have 1,000 formats, but like if it's 5 or 6 or 7 or 10 or 12, I don't care.
So since the standards topic came up, so there is through the IFS standards efforts, there is a 35 member effort to develop terminologies to agree upon basically what is AtomProbe, for example, or define it properly, and then send it to things like ISO TC 201 standardized terminology listing for different techniques and things like that. So one thing which I wanted to ask all of you is, are there specific terms that might be worth, or that you from the technical committee might be able to define? Because terminology is kind of the first starting point for developing standards for any of the analytical techniques. So have you guys thought of, I mean, if there is something specifically, any specific analytical or database terms that we use commonly in AtomProbe can be defined by technical committee, that's something which we can incorporate into the overall AtomProbe standards efforts and eventually take it to something like ISO.
It goes to the question of metadata definitions and eventually defining, at least more or less formally, what is the connection between different objects of the terminology. And that's something that we have discussed in the TCs, but I think it's pretty at the start. And I think that is a great motivation from you, Aaron, that we should keep up on that, because personally I can only agree. If you think about how things and terms are connected together and how this relates to workflows, then you might come up easier with an understanding and a more abstract understanding of what AtomProbe is and how the individual workflows, which seemingly are all somewhat different, however, by the end of the day are quite related and nothing but specific specializations of basically, by the end of the day, very similar workflows. Yep. And I think that is a question that at least I will raise strongly in the next round in the APTC. And I would be quite happy if also there's interest in the community, because I think this is a key ingredient that can help us define file formats, because otherwise somebody makes a choice what you place there, which type of metadata. Of course, you can pass in between them fully on that page with what Peter's saying. But how can we make this even more efficient? How can we combine this? How can we couple this with the individual ideas and the workflows?
Yeah. So the people who do this for a living, in terms of metadata and FAIR and everything, they talk about subject-specific ontologies. So this is where we as Atom Probe experts can talk to someone who basically runs a database for a living and say, these are the things which are important. This is where we can inject our Atom Probe knowledge to kind of empower the data and to be able to make it, more widely usable, and so people can understand what it is, even if they're not necessarily an Atom Probe expert from the start.
And maybe let me jump in there. So as I said before, in our case, the push didn't come out of Atom Probe necessarily, but out of collaborative research centers with very, very different types of data. So I think what we really need to make sure is that we are as Atom Probe unspecific as possible. Yeah? Because there's a lot of metadata which is absolutely not Atom Probe specific. Date of experiment, person carrying out the experiment, name of the specimen, none of this is Atom Probe specific. And we need to really make sure that we're as interoperable with any other data as we can. And that the metadata definitions are as human readable as possible. So be verbose, use long names. Yeah? Nothing should be called APT. Nothing should ever be called APT. Nothing should be called FIB. Nothing should be called SEM. Okay, because if I give that to my friend over at chemistry, they don't know what that is.
And yeah, I'm actually seeing even in the ISO terminology definitions, there are a lot of terms that are already defined for other techniques. So like mass to charge ratio, signal to noise ratio, or background. All of these generic terms are actually intertransferable between different techniques. So I would assume there are things that are in the standard literature out there that can be generalizable. I agree with you, Peter.
Yeah. So there used to be a site hosted on NIST.gov called APT nomenclature. My link to that doesn't work anymore. Hopefully someone knows where that's moved to, but that specifically was a library of that kind of Atom Probe specific nomenclature.
That came out of the MSA Atom Probe FIG several years ago. Yeah, and it has been down for some reason. I've been bugging Fred and Eric Steele to get it back up online. But we are now trying to borrow some of the definitions there and then try to debate it in the community, which is about 36 or 40 of us from many different countries. Now, the problem is even defining Atom Probe, the term Atom Probe itself took us, I believe, like 100 emails or 200 emails back and forth. So it's taking a lot of debate, but I think the focused smaller number team in here might be able to really churn out some good definitions for maybe specific metadata terms, or even analysis-based things like ISO surface for ISO concentration or ISO composition surfaces, or proxy grams and things like that, which are, everybody uses it, but there is no standardized definition. So that might be something worth considering eventually. Yeah.
That's great. Thank you very much.
I'd like to just quickly piggyback on that. I think one of the things we're trying to do with the Atom Probe standard file format is to try and leverage these definitions that are out there. And what we're trying to do is make it so that it's not, the file format is basically versioned. And so we can reintegrate community definitions back into that standard, such that we are tightening how we define and how we use these various fields. And so software will need to evolve to be able to handle these tighter and tighter definitions through time. At the moment, we're being quite broad, just because, as you say, it is actually quite a difficult effort to get everybody to agree on certain things. And so we are being a little bit lax with some of our terminology, but I think, if once the standard is a bit more widely available, that might have food for thought that can go into these ontology definitions. They can then be fed back into the standard once everyone's got an agreement on that. And that can be a rolling effort going forwards.
Yeah, so I don't want to cut the discussion short, but we have reached our time, and I'm aware people will have other meetings. People in Sydney may want to go to bed if it's 4 o'clock for you. We very much hope that these kind of discussions can go on. we don't have really a good forum for doing that. There is the IFES Atom Probe Facebook page. Feel free to post there. Contact your local TC representative or use the email address. We'd love to have more input for our discussion, have more people to talk to. I would just like to say thank you to all the people presenting. I think it was great to have so much content for this remote meeting, and I think it's gone very well. I hope you agree. Like I said before, we will be in touch with that list of software and contacts, the recordings, so you've got everything for your records. Yeah, if you've got more questions, we'd love to hear them, and hopefully we can get some more responses as well. And with that, I'm going to leave. I think I can leave the webinar running if anyone else wants to post in the chat or have a discussion. You're free to do that. I'm not entirely sure whether I can leave as the host and the meeting continue, but I'm going to stop the recording there. And yes, enjoy the rest of your day, morning, evening, wherever you are.
Okay.
Thank you, everyone, and have a good morning or afternoon or whatever you guys got. Good day, everyone. Bye.
Great. Bye-bye.
Thank you. So this is the point where we can see which attendees were actually listening by who's just going to hang on for as long as the.
Yeah. Yeah. But I'll drop out now as well, because I know I need to spend another 1.5 hours for transcoding the Zoom file.
Yeah. I think it overall very well. I think everyone did a good job.
Yeah, thank you, David. So I think there's pretty much interest also in that community, and we should redo this again at some point.
And hopefully we can have another session in person next year at APTNM.
Yeah, hopefully, yeah, hopefully.
Or else maybe the rest of the world can, and the US will still be under no travel allowed, given how it works.
So we need to keep our masks on.
Yeah.
And be rigorous. Okay, so I also drop out. You guys have an excellent day or night, wherever you are listening to it. And thank you very much for joining the channel. If you have questions, if you have comments, and that was mind-inspiring, discuss with your colleagues, send us an email. And with that, stay safe. That's the most important thing of all. Bye-bye.
Bye.